Recording in progress. National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation No. 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development, and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
menunggu kita conference room.
Good morning, ladies and gentlemen. I hope that you are well. So before we begin the event, I would like to read the conference guide. So the host, could you please try to share screen the conference guide? All right, so allow me to read the conference guideline. So for presenters and participants are expected to join at least 30 minutes before the conference is started, and all participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom username according to the following formats. For presenter, you could use code of abstract or paper underscores name underscores institution. And for non-presenters, we could use the format P underscore name underscore institution. And please make sure to mute the microphone, especially during the presentation of keynote speakers. And in the parallel session, presenters should activate the camera. And questions from all participants are delivered using the raise hand, Q&A, or chat feature in Zoom. And when Q&A or chat feature selected, please deliver questions according to the format name / institution /to name of the speaker and /questions. And every session will be led by a moderator. Those moderator will decide which questions is to be discussed or to be answered. And all participants should fill the list of attendees via the link given in each session and abide all the rules listed on the screen. So now we're waiting for Dr. Iman Hidayat to join us. Thank you.
National Re- National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Ladies and gentlemen, may I have your attention? We would like to initiate the symposium in a few moments. The Honorable Dr. Laksana Trihandoko, Chairman of National Research and Innovation Agency, BRIN Indonesia. Honorable Dr. Iman Hidayat, Chairman of Research Organization for Life Sciences, BRIN. Dr. Rati Asmananingrum, Head of Research Center for Biotechnology, BRIN. Head of Research Centers and Head of Bureaus in BRIN, distinguished speakers and moderators, representatives from ministries, institutions, and universities, and all distinguished participants. Good morning. It's a great pleasure for me to welcome every one of you to the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBIO 2021. Global Platform on Biodiversity and Biotechnology. ISIBIO 2021 is a continuation of the previous 7 seminars, first held in 2012, and has been annually carried out by the Research Center for Biotechnology, BRIN. This year, the symposium is held in collaboration with Indonesia Biotechnology Consortium, or IBC. The objective of ISIBIO 2021 is to provide a platform for academic dialogue on biodiversity and biotechnology, and for the exchange of recent updates on biodiversity and biotechnological researches. The International Symposium is conducted virtually live from Zoom meeting and official YouTube channel BRIN. Now, before we begin, allow me to read the conference guide. Presenters and participants are expected to join at least 30 minutes before the conference is started. All participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom according to the format. For presenter, code of abstract or paper _name_institution. For non-presenters, you could use P_name_institution. And please make sure to mute the microphone, especially during the presentation of keynote speakers. And in parallel session, presenter should activate the camera. Questions from all participants are delivered using raise hand, Q&A, or chat feature in Zoom. And please deliver the questions according to the format, name/institution/2, name of the speaker, and /questions. And every session will be led by a moderator, and all participants should fill the list of attendees via the link and abide all the rules listed on the screen. Ladies and gentlemen, please be prepared and remain seated. We will sing to national anthem, Indonesia Raya. Ladies and gentlemen, we would like to begin the International Symposium with welcome remarks from the Chairman of Organizing Committee ISIBIO 2021. Please welcome Dr. ISIBIO.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Chairman of National Research and Innovation Agency, Dr. Laksana Trihandoko, the Honorable Chairman of Research Institution for Life Science, National Research and Innovation Agency, Dr. Iman Hidayat, the Honorable Director of Research Center for Biotechnology, National Research and Innovation Agency, Dr. Rati Asmananingrum, the Honorable President of Asian Federation on Biotechnology, Professor Dr. Duk J.O., the Honorable President of Indonesian Biotechnology Consortium, Dr. Mastur, all of our invited speakers from Indonesia, Malaysia, Japan, Saudi Arabia, Netherlands, and USA. Also, to our distinguished presenters, participants, and invited guests. Good morning, welcome, and thank you for being here with us today. Ladies and gentlemen, it is my pleasure to welcome all of you at the beginning of these two events. First, the 8th International Symposium of Innovative Bioproduction Indonesia, or ISIBIO 8 2021. And second, the 7th Indonesian Biotechnology Conference, or IBC 7 2021. Today, it marks our first event, ISIBIO 8 2021. It has been 8 joyful years that we host this annual event. And this year is special because ISIBIO 8 2021 is held in conjunction with IBC 7 2021. This would not be possible without a collaboration between Research Center for Biotechnology and Indonesian Biotechnology Consortium. So, in this opportunity, I would also like to acknowledge and appreciate the Indonesian Biotechnology Consortium and the team for making such a great collaboration. Distinguished guests, participants, ladies and gentlemen, this year, ISIBIO at 2021 has received high appreciation from 107 attendants who represent science, education, and industry communities, including 76 oral presenters, which 65 of them are paper presenters from various institutions and universities in Indonesia and Malaysia. As the output of this symposium, all accepted papers will be published in an international proceeding, IEP conference proceeding, and an international journal indexed by Scopus, Indonesian Journal of Biotechnology. On the third day of the event, IBC 7 2021 will provide you with 2 attractive talk shows. First, we will hear recent updates and the future of biotechnology startup companies in Indonesia from several CEOs of biotech startup companies, including Acumen Research Laboratories, Algae Park, and Nusantix. Secondly, the talk show will discuss program Merdeka Belajar, Campus Merdeka, in order to share information to us about the opportunity of this national program in the field of biotechnology and biodiversity research. As the chairman of ISIBIO 8 2021, I would say, please make the best of this opportunity and enjoy casual yet meaningful scientific discussion with the experts on a wide range of topics. Just like the theme of this event, Global Platform on Biotechnology and Biodiversity, we expect that these events can act as a bridge for knowledge and technology sharing, and also an initial platform to spark research collaboration in the field of biotechnology and biodiversity. Last but not least, I would like to acknowledge all the sponsors who support ISIBIO 8 and ISIBIO 7, IBC 7. Tuwadia Prima Mulia, Elo Karsa Utama, Pandu Bioscience, Gitek Jaya, Nusantix, Merck, Crop Life, ITS Science Indonesia, Science Work, Gincraft, Biosense Medica, Berka Niaga Medica, and Nutrilab Pratama. Thank you for the contribution and support for the events. We hope that these mutual partnerships will continue in the years ahead. To end my remarks, I once again would like to express my gratitude to all of you for your participation and contribution in this symposium. Thank you very much for your kind attention, and please enjoy the events. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Bapak Asep, for the warm welcome remarks. Ladies and gentlemen, next we will hear welcome remarks from the head of Research Center for Biotechnology BRIN. Please welcome Dr. Rati Asmananingrum.
Thank you. Honorable Chairman of Organization for Life Sciences, National Research and Innovation Agency, Dr. Iman Hidayat, Head of Research Center of Organization for Life Sciences, National Research and Innovation Agency, President of Indonesian Consortium on Biotechnology, honorable speakers, honorable moderators, honorable invited guests, distinguished ladies and gentlemen. A very good morning to all of you. It is my great pleasure to address at the opening of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2021 in conjunction with the 7th International Biotechnology Conference 2021. This year, the symposium and conference have the theme The Global Platform on Biodiversity and Biotechnology. The term biodiversity refers to the variety of life on Earth at all its levels, from genes to ecosystem, and can encompass the evolutionary, ecological, and cultural process that sustain life. Biodiversity is very crucial to our life for many reasons, from economic, ecological life support, recreation, cultural, to scientific. Any loss or deterioration in the condition of biodiversity can compromise all of the value and affect human well-being. Distinguished ladies and gentlemen, we are currently experiencing very serious problems related to climate change. It is one of the biggest challenges of our times. It is already happening. Temperatures are rising, drought and wildfire are starting to occur more frequently, rainfall patterns are shifting, glacier and snow are melting, global mean sea level is rising, land degradation is occurring, and biodiversity is decreasing rapidly. All the effects due to climate change have affected two important aspects of our life, food and health. Distinguished ladies and gentlemen, nearly 200 nations reached a climate agreement on COP26 to the role of fossil fuel in the climate crisis. The world needs to cut the rate of the greenhouse gas emission by almost 27 billion metric tons a year in order to limit global warming to 1.5 degrees Celsius by 2030. This effort certainly requires hard work globally and takes quite a long time. We certainly need a strategy on how to adapt and be ready to face climate change. Indonesia is the top 3 of mega biodiversity country, but the problem is that the use of biodiversity is still limited to provide solution to the nation problems, especially to encounter climate change. Distinguished ladies and gentlemen, biotechnology is technology that utilizes biological system, living organism, or part of this to develop or create different products. Research Center for Biotechnology, National Research and Innovation Agency is trying to continue to play an active role in utilizing biodiversity to answer the nation's problem. As a research center that has competence in health, food, and industrial biotechnology, we have carried out various research activities for food security and health. Several research activities for food security are development of stress-resistant and nutrient-rich transgenic rice and soybean, nutrient-rich and long shelf-life cassava, superior seeds cattle, probiotic-based feed for livestock health, and so on. In health security, we are discovering the active compound for medicine from microorganisms, plants, and animals. We also developing the biological-based product for infectious and degenerative disease. Distinguished ladies and gentlemen, this seminar and conference is held as an effort for all of us to share our competencies and experiences so that we can collaborate and work together to obtain output and outcome that can answer existing and future problems. We all hope that we can play an active role as part of the world to create a better life. Before I close my remarks, I would like to thank to the organizer committee for their hard work and valuable contribution to this seminar. And my deepest gratitude also goes to the Honorable Head of Organization for Life Sciences, National Research and Innovation Agency, honorable speakers and moderator, the sponsor for their great contribution, Indonesian Consortium on Biotechnology, and all of participants who attend this event. So as a representative of the organizer of the 8th ISIBIO and the 7th IBC, I officially open this event. Welcome to the symposium and conference. Please enjoy the scientific knowledge sharing, global platform on biodiversity and biotechnology. Thank you.
Thank you, Ibu Rati, for the wonderful remarks. Ladies and gentlemen, for the next, we will have a keynote speech by the Chairman of Research Organization for Life Sciences, BRIN, Dr. Iman Hidayat, and which is in this occasion representing the Chairman of BRIN. Good morning, Bapak Iman.
Good morning, Moderator.
May I
share the presentation?
Before that, I would like to read a CV for Bapak Iman. Is that okay?
Yeah.
All right. So before we begin, allow me to read a short profile of Bapak Iman. Dr. Iman Hidayat earned his doctoral degree in plant pathology, Faculty of Agriculture, Chiang Mai University, Thailand. and he is currently the chairman of Research Organization for Life Sciences BRIN, and previously he was the director of Research Center for Biomaterials, Indonesian Institute of Sciences LIPI. His research interests include development of advanced material technology based on plant lignocellulose and microbial shipping and cellulose. So without further ado, with Mr. Iman Hidayat, the time is yours.
Okay, thank you very much, moderator. I will try to share my PowerPoint first. Good morning, ladies and gentlemen, distinguished participant, keynote speaker, and all participants of the 8th ISIBIO and 7th IBC conference. I hope that all of you always in the good health in current condition and the pandemic COVID-19 condition, so that we cannot meet each other in this great opportunity. But we still can share our knowledge in the current conference. We can contribute to each other about our progress of research. Let me allow me to represent the chairman of National Research and Innovation Agency BRIN, Pak Dr. Laksamana Trihandoko, to deliver policy on global platform on biodiversity and biotechnology research and innovation of BRIN. First of all, I would like to congratulate, of course, to the Research Center for Biotechnology BRIN for the ISIBIO 8 today. And also I would like to congratulate the Consortium of Biotechnology, Indonesian Consortium for Biotechnology, for the 7 IBC. In today's presentation, I will show several policy of BRIN in managing biodiversity, as well as in creating some strategy in biotechnology research and innovation to accelerate the contribution of research and innovation to the national economic development. We all know that Indonesia is a country with a huge biodiversity, from terrestrial to marine to freshwater biodiversity, from animal, plant, microorganism. This huge biodiversity is advantage or basic capital for Indonesia actually to develop economy that very important for Indonesia to develop the economy, because the biodiversity, at least it has economical value, it also ecological value, and also it has cultural value. So it is important for us as a researcher and academic to conserve as well as to utilize the biodiversity. However, in managing biodiversity, it's not easy. Many countries with huge biodiversity belong to developing or poor countries. I don't know why, but because the managing biodiversity is not easy. We, how can I say? We have to maintain, we have to unreveal the biodiversity, we have to develop the technology to utilize. In the same time, The growth of human population also become problematic for, especially for Indonesia, to manage the biodiversity. Growing of population, it means growing in food demand. It means it will need more area for them. It means growing for industrialization. At the same time, the world climate change also increasing. It is, how can I say? It's not easy to managing biodiversity. So in managing biodiversity, at least there are two parts. The first one is how to conserve the biodiversity for future, and then how to utilize the biodiversity for economical development. BRIN has the responsibility in Indonesia to carry out both function, conservation and utilization. For conservation, it is the responsibility of BRIN to provide ex situ conservation. In the ex situ conservation, BRIN provides the biodiversity exploration and inventory program, human resources, as well as supporting funds. And BRIN also provide the facility for the national depository of plant, animal, and microorganisms. And also BRIN responsible for all stakeholders in Indonesia, not only Indonesia, in other countries as well, to access all the biological collection and information preserved in culture collection, herbarium, and museum managed by BRIN. And BRIN also provides services for the identification of biological diversity. For the plant conservation, ex situ plant conservation, BRIN also developed national botanical garden. Until now, there are 4 national botanical garden, Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Garden, and Bedugul Bali Botanical Garden. Next year, BRIN will develop more national botanical garden, at least 5 more national botanical garden. And in the same time, BRIN collaborate with local government to develop local botanical garden. It is important because with the accelerating of industrialization and also climate change, global climate change, many plants is now going to extinct. It is the responsibility of BRIN how to conserve the plant before extinct. In the utilization, there are also several examples of BRIN to develop a low-level technology and also high-level technology. So back to the biotechnology, there are many definitions, but one of the simple definition is the manipulation of living organisms or their component to produce useful product. So biotechnology is the key for the economical development based on biodiversity resources. It can be for bioenergy, medicine, veterinary, agriculture, fine chemical products, and pharmaceutical. It is challenge for academicians and researchers in Indonesia how we can deliver all of these challenges. Where are we now? We know each other. Where are we now? Do we have this product until now or not? It is the challenge for us. So the current condition, We know that biodiversity is theoretically the capital for economical development. And it is the great advantage for Indonesia. And the tool to make it is biotechnology. Unfortunately, until now, we still left behind to other countries, and we has not taken full advantage of this potential. We conduct several biotechnological research and several products such as superior variety of staple food, paddy rice, cassava, and also we do research how to develop superior livestock from local varieties, and also we develop vaccine now, as well as how to develop various vaccine, not only SARS-CoV. But the challenge is not only that, because this biotechnology is the key for food security and health resilience. As Dr. Rati previously mentioned, biotechnology is the key to nation food security and health resilience. But we witness now, until now, that Indonesian national health resilience is still weak and still not meet the demand. I can show you the case of study for the COVID-19 case. We know that the first case of COVID-19 is in November 2019 in Hubei. The first genome or SARS-CoV was announced in January 2020, and Indonesian first case of COVID in March 2020. Since the first cases, around 6 months from the first cases in Hubei, Sinovac already conducted phase 1 and 2 human clinical trial. And they get the result in June 2020. And Indonesia first administered of COVID-19 vaccine was conducted in January 2021. Until now, already about 46% of Indonesian population has received vaccine of COVID-19. Compare to the Chinese vaccine or Chinese biotechnological research, Merah Putih vaccine have just finished preclinical trial on macaque last month. So we know that there is huge gap between China biotechnological and Indonesian biotechnological. I will show you several bottleneck or constraint in the development of vaccine Merah Putih, in Indonesia. So there are several technology to develop a vaccine, inactivated virus from the viral vector and so on. But the bottleneck is Indonesian researchers and academicians have never developed vaccine. So in terms of experience, skill, we are left behind. The second one, collaboration among researchers, academicians, and industry is also need to be improved, something like that. And the last one is infrastructure. To develop vaccine in Indonesia, we don't have GMP facility. We don't have certificated animal Basel III facility. So it become a great problem for us in the pandemic, how to develop the vaccine. To answer this problem, so BRIN start to develop infrastructure and then how to develop human resources, as well as how to develop the research and innovation environment in Indonesia. Now BRIN developed several facilities in Indonesia, not only in Cibinong, to support the research and technology to utilize biodiversity in Indonesia. This is the building of Indonesian Biodiversity Collection for and data center, and the building will be operated in August next year. And this is the Genomic Biodiversity and Environmental Laboratory building to conduct research on genomics, protein engineering, cell development, cell manipulation, and so on. It will be operated in January 2022. And also BRIN also have BSL3 facility to develop infectious research, disease research. And also BRIN also build the integrated laboratory of bioproduct in Cibinong. All of these facilities are in Cibinong. This not and BRIN also build some facility not only in Cibinong but also in other region in Indonesia, including in West Java, in Sumatra, in Ambon, in Lombok, West Nusa Tenggara, and so on. All are open access for all stakeholders. So not only researcher for BRIN, not only researcher from BRIN that can access all the facility, but other stakeholders, academicians, industry also have the opportunity to access and to utilize the infrastructure through ELSA system. So BRIN developed the system so that all stakeholders can access and can use the facilities. In terms of researcher and industry relationship, BRIN also developed the platform how industry can get involved in developing products. Until now, in Indonesia at least, researcher or research institute in Indonesia doing their research without involving industry. So at the end, mostly the product do not meet the industrial standard or demand. It become chronic problem in Indonesia. Therefore, BRIN now start to involve the industry from the research planning until how can I say, from the planning until the execution, the research involve the industrial resources. So BRIN in this context as a multi-stakeholder collaboration hub, it is not only involving industrial stakeholder but also other stakeholders. To develop human resources, BRIN provides several platforms. For example, visiting researcher assistant researcher, research assistant, and visiting scholar. It is important not only for BRIN, but also for other stakeholders to develop the mutual relationship. For example, in the research assistant, student that conducted research at BRIN has the opportunity to become research assistant and then has the opportunity to continue their study full funded by BRIN through by research program. So it is a great opportunity for students in Indonesia as well as for academicians in Indonesia. Not only that, but also to other stakeholders, so they can increase their competency, knowledge through the system. So BRIN will facilitate research partnership, stimulate multi-stakeholder collaboration, and stimulate the innopreneurship. And we hope that for the next 3 or 5 years, we can harvest the output from this program. I think that's the presentation that I can deliver regarding the policy of BRIN to accelerate biotechnology, research and innovation in Indonesia. Thank you very much.
Thank you, Bapak Iman, for the very insightful speech. Ladies and gentlemen, before continuing to the next agenda, we will have a photo session with all participants. So could you please kindly open your video and the host will help us to take a picture. All right, so there are 7 slides. We will start with the first slides. For the photo, could you please try to give your best smile in this fine morning? Okay, so we'll start for the first slide. 1, 2, 3. The second slide. 1, 2, 3. The third slide. 1, 2, 3. The fourth slide. 1, 2, 3. The 5th slide. 1, 2, 3. The 6th slide. 1, 2, 3. And the last slide. 1, 2, 3. Thank you, ladies and gentlemen, for your participation. Distinguished guests, ladies and gentlemen, we will continue to our next agenda, which is plenary session with speakers Professor Junichi Tanaka from Department of Chemistry, Biology, and Marine Science, University of Ryukyu, Japan, and Dr. Mastur, President of Indonesian Biotechnology Consortium, or KBI. This session will be moderated by Professor Ekowati Hasana. And before we begin, allow me to read a brief profile of Professor Ekowati. Professor Ekowati Hasana achieved a doctoral degree in Food Science Program, Biotechnology, IPB University, and master's degree in Department of Food Science, Nutrition, University of Rhode Island, and for Bachelor, Agriculture Technology, Universitas Gajah Mada. She is currently a professor in Research Center for Marine and Fishery Product Processing and Biotechnology, Ministry of Marine and Fisheries, Indonesia. Professor Ekowati has been conducting researches in the area of marine biotechnology and fisheries product. Now, without further ado, Professor Ekowati, time is yours to lead the session. Good morning, Professor Ekowati.
Okay, yeah. Now, ready. Thank you, Mbak Wiwit. Assalamualaikum warahmatullahi wabarakatuh. Honorable speakers, distinguished guests, and all participants, good morning. Welcome to the plenary session of the 8th ISIBIO 2021, and in conjunction with the 7th Indonesian Biotechnology Conference, IBC. In the next 1.5 hours, I'll be the host of this session, serving as your moderator. Today, we will be hearing presentation from our speakers, Professor Junichi Tanaka from Japan and Bapak Insinyur Mastur MSc PhD from Indonesia on this very timely subject. But before we get started, please allow me to introduce our speakers. Professor Junichi Tanaka is a professor at the Department of Chemistry, Biology, and Marine Science, University of Ryukyu, Japan. He has been attached with the university since 1996 as instructor and as associate professor in 2007, and finally appointed as professor in the University of Ryukyu since April 2007. He is also alumni of the University of Ryukyu for his master and his bachelor degree. And he achieved his doctoral degree from Pharmaceutical Science, Osaka University. His research interest is on new bioactive compounds from coral reef life, particularly invertebrates. He also engaged in collaborative research with other researchers on related research such as mode of action, structure, activity, relationship, biodiversity, and biosynthetic genes. He has a lot of experience working abroad as a visiting scientist. In 1991, he was visiting scientist at Chulalongkorn University, Thailand, and in 1993 and 1995 at Parmamar, Spain, and in 1997 he was with LIPI Oceanology Indonesia, and in 1998-2007 he was with Max Planck Institute for Biochemistry and Saarland University in Germany in 1998 and 2007. Now, please welcome Professor Junichi Tanaka, who will be present marine natural products and biodiversity of marine organisms. Prof. Tanaka, are you there?
Yes, thank you.
Yes, your time will be 40 minutes,
Okay.
Professor. Okay, time is yours now.
Okay. Thank you very much for the kind introduction, Professor Ecology, Hassana. Let me upload my slide. Okay. Do you hear me? And also, do you can you look at my slide?
Yes.
Okay.
Yes.
Okay. Let me start. Okay. I'd like to thank the organizing committee for inviting me as a plenary lecturer today, and also my former student, Dr. Penny Amari, for arranging this opportunity. It's a great honor for me. Okay, before talking on the contents, let me introduce where I am based on. if you compare Okinawa from Indonesia, it's so small. Okinawa is located at southwest part of Japan and it consists around only 60 islands or so. And it is located east of Taiwan.
Sorry, Prof. Tanaka.
Any problem?
Could you make your voice loud? Could you make your voice louder?
Okay, I got
Louder,
it.
please.
Is it okay now?
Yes. Yeah, it's better.
Okay, sorry. My voice is usually small. Anyhow, let me continue my talk. Okay. Anyhow, I am from this island, and the island of Okinawa is around. It spans around 500 kilometers in distance. Anyhow, thanks to the warm cross current in the Pacific, the islands are washed by warm water. So, just like as in Indonesia, the islands are surrounded by coral reefs. Our university is located on one of the island, largest island, and these are our buildings. And it is located at a nice place to work on chemical and biological aspects of coral reef organisms. Okay. Today, I'd like to talk about marine nature product and some of the diversity work we do. But after the brief introduction on marine nature product chemistry, I'd like to talk about some typical natural product studies, including an example of structural illustration or determination. Some of you may be not familiar with chemical analysis, so please, but it's just part of the basic matter for our field. And we usually look for newer molecules from marine sources, but even a normal natural product, there is some application. I'd like to show one example. And during natural product study, sometimes you may need to be careful on the research. And I'd like to show one process example. As a second topic, I'd like to show some bioresources in nature and also a phenomena of some marine organisms. And so I'd like to show some useful or precious molecules from marine organisms, and also some unique phenomena of metabolites and organisms. And at the last, I'd like to talk about chemical and genetic diversity of marine organisms, of two groups of organisms. These are sarcophyton, soft corals, and Iridia nutrients. Okay. All right. Natural products are, of course, metabolites produced by organisms. Plants have been utilized from recent time, but microbial metabolites may be utilized from middle 20th century. Compared from then, marine sources, marine organisms are relatively new, maybe utilized after, mainly after 1970. One of the pioneers is late Professor Sawyer of the University of Hawaii, and he was dubbed as the father of marine natural products. And why are we attracted by marine organisms? The answers may be the huge number of species in the water. Once it was expected that more than 500,000 species, but some paper reported this estimate, but every year the number is increasing. So I'm not sure the exact number of species anyhow. There is a very huge amount of huge number of organisms in the water. Not only sessile marine organisms, but there are also marine microorganisms. And of course, we can expect the environment is totally different from terrestrial area. Apparently, it is aquatic environment. They operate with salinity and pressure, etc. So, after the long evolutionary history of new organisms, they developed different strategies from that of terrestrial organisms. And also there are a number of relationships between organisms. Some are probably parasitic, symbiotic, etc. So, yeah, probably it's endless target. Okay, let me talk about some basics of my method of study with our new example. Okay. First is typical structure determination work, because we are dealing with chemicals, so without knowing what kind of molecules we have, there is, we cannot proceed further. Okay. So currently, a group of compounds originally reported by Australian workers. And when we worked on a cytotoxic constituent of this sponge in Okinawa, my student isolated two new molecules. These are stereoisomers, or diastereomers to the known ones. One of them, named as Franklin X, is a non-crystalline molecule, but after many trials, my student found that its derivative, having bio in the structure, can be crystallized. And by using X-ray crystallography with diffractometer, you can reduce the whole stereochemistry. With X-ray diffraction study, usually nobody claims that your conclusion is wrong or no. But when you do crystallography, it totally depends on the nature of the molecule, whether it is crystalline or not. In case of, as I told, in case of Franklin X, the original material was not crystalline. And according to Professor Fujita of University of Tokyo in Japan, it is a 100-year-old problem whether the molecule is crystalline or not. Even now, we cannot predict which molecule will crystallize or not. But he invented a so-called crystalline sponge method to solve non-crystalline molecule by crystallography. I heard that currently it can be applied only to small molecules, but hopefully his group invents a molecular scaffold to solve various molecules in future. Yeah, it is developing. Okay, another molecule named Franklin Y was isolated together with Franklin X in the previous slide. In this case, we could not find any crystalline derivatives. So we have to solve the stereochemistry with NMR instrument as well as most conventional natural product studies. But in this case, although it seems to be clear on the spectrum, but there are some signal overlaps, and we face a little bit difficulty on assigning the stereochemistry. So once again, my students try to make several derivatives, and one of them having alcohol at this position instead of double bond, and plus doing so-called NOE study and finding out the stereochemistry. Those are the typical works. But in another case, it was not so easy. When we worked on a cultured microalgal metabolites for antiviral screening, we isolated a new molecule named caveolin in relation to a known molecule, protein. Protein was already reported as a cytosol constituent, but we also reported its crystalline structure. But in case of caveolin, not like protein, it was not crystalline. So we used the NMR to illustrate the whole stereochemistry. But with NMR study and additional works, we can only find that 5 partial stereochemical structure colored in differently. So the final molecule must be one of the possible structures. So in this case, I asked my colleague, calculation chemist, to estimate optical rotation value to compare with the actual observed value. So my colleague generated 16 possible structures, and after calculation, he found stable conformals. Then he calculated optical rotation value. And in our final estimate, the whole stereochemistry may be like this, but it is just estimate. In future, I hope synthesis chemists may conclude whether our tentative structure is correct or not. Okay, second topic is the application of a non-natural product. Polychondramide is a member of trisoxazole macrolide, having 3 consecutive oxazole moiety in the macrolide. This member of molecule was discovered only from mine sources. And they are graphylide, caveolamide, and polychondramide, and microlide, and their congeners. And their mode of action or cytotoxicity or antifungal activity was caused by targeting site actin, the most abundant protein in our body. And with my collaborators, we reported how the macrolide bind to the target protein actin, and also we could solve the whole stereochemistry by crystallography with protein. And we also understood the mode of action. Okay, although the structure is quite complex, we can obtain the pure compound of this molecule from natural sources in gram quantity. And this situation is quite different from some other members, some such as acrylamide, which are quite complex as a mixture of similar molecules. So we use for some other purpose. In addition, the molecule contains enol moiety, which is a good target for our nucleophilic reagent. So we made several drips based on these molecules. At first, we prepared simple diptych named the ISIBIO A and its ephemer by treating iconbramide with ammonia water. By evaluating cytotoxicity against cells, culture cells, and also affinity to fluorescent GRP, it seems to be that the ISIBIO and ephemer has a higher affinity than original iconbramide. Second application was to make a fluorescent probe. As we know that this class of molecule binds to the leading edge of actin fibers in cells, we plan to stain the peripheral region of cells. By attaching fluorescent moiety through shorter or longer linker moiety, they stain cells like as shown, as in the right. Okay. The last application was making affinity gel. Nowadays, molecular biology methods are used for finding the amino acid sequence of proteins, but I wonder if there might be an opportunity to isolate actin-like proteins. In this case, affinity gel with shorter linker did not work, but with a longer linker moiety, the gel is likely to work. Okay, in the third topic on natural products, I'd like to discuss about artifact formation. When you isolate molecules from natural sources, you may believe that they are natural, but it's not. There are quite a number of possibilities to isolate non-natural molecules, namely artifacts. Here I listed causes to make artifacts. 3 example molecules are shown. The first one is acetone adduct after aldol-like reaction. Second one is hydroquinone, because as you know, it's easy to deoxide to quinone. And the last one is contains a lactone moiety, but under mild acidic condition, it cleaves into carboxylic acid and alcohol. Okay. So it totally depends on the structure and the conditions when you handle these molecules. In the papers published by my neighbor laboratory, working on my models as well as ours, they reported some molecules with hydroperoxide moiety. These are hydroperoxide. As a hydroperoxide signal in NMR is conspicuous, appearing chemical shift 7 to 7.5. So we believe that we did not look over it. However, we have never encountered molecules with this functionality. When we think about the difference between the 2 laboratory conditions, there is a significant difference. Our neighbor laboratory has a big windows facing south. That means we are located in the northern hemisphere. Sunlight penetrates through windows, and they don't use window shade. In our laboratory, our windows are tree shaded for the southern south part, and the windows are not facing north. So we examine the possibility of sunlight. It is already known that singlet oxygen will be formed from normal triplet oxygen, which we breathe, not directly with the light, but indirectly through excited photosynthesis fiber or pigment. As most of extracts from natural sources contain pigments, so it might mean that singlet oxygen may be formed if solution of the material is exposed to sunlight. And in addition, it is a well-known phenomenon that singlet oxygen will react with a molecule containing double bond to give hydroperoxide through in reaction, as shown in this scheme. Compound 1 is a stable marine natural product from a sponge, and we kept it in a freezer for 3 decades and used it as a model molecule. We incubated in compound 1 with artificial pigment named rose bengal, with or without sunlight. And of course, without sunlight, there's nothing happens. But as it is expected, and if it is exposed to sunlight, hydroperoxide, compound 2 was formed. But when we left the mixture for 2 weeks by the window, it resulted in the isolation of further oxidized or degraded molecules 3 to 5. Particularly, compound 5 is highly oxidized to incorporate the peroxide in the molecule, which is a little bit similar to the known anti-malarial drug artemisinin. With conventional oxidizing agent, I believe it is difficult to oxidize position like here. So it might be useful to make some unusual derivative, though the speed, reaction speed is very slow. Okay, the formation of compound 2 and 3 can be explained as in this scheme. The air reaction product 2 will react with water, namely Fock reaction, to give hemichemical compound 6. This compound will flip to give compound 3 tetraldehyde. But for other products 4 and 5, it is not easy to explain how they are formed. After publishing our results early this year, we noticed that enantiomers of compound 1, 2, and 5 were already reported by Thai researchers as sponge metabolites. On the other hand, Israel group reported another similar molecule named axiprins as sponge metabolite. I don't know their laboratory conditions, but we have to be careful when you handle the material under sunlight. Okay, I summarize the first part of my talk. When you work on structures of natural products, suitable methods should be chosen for determination or elucidation. We usually neglect or ignore non-natural products, but even non-molecules can be used for some purpose. And of course, as I thought, it is better to avoid sunlight to avoid artifact formation. But if you want to have some unusual oxidizing metabolite, singlet oxygen might be useful. Right. For the second part of my talk, Now, I like to, in this section, first I'd like to show a few cases of bioactive metabolites from and their sources from Indonesian water. In the latter part, example of the same or similar compound from different organisms are shown. As I worked on some marine resources with my collaborator in Indonesia and Japan, I like to show some resource organisms. Jaspermide or just platinolite, one compound has two names because they were different. They were reported by two groups independently. Anyhow, this kind of molecule is a DEPS peptide, so-called DEPS peptide, and showing active polymerization activity. And it is sold as a life science reagent with a good price. This source sponge spread from the Philippines to Indonesia, and you may supply the molecule, though isolation is a little bit difficult. Zesta sponge C is a unique alkaloid molecule and sold as a reagent as an IP3 receptor antagonist. This sponge distributes from Okinawa to Indonesia, so widespread. The problem is purification rates. And there are many similar alkaloids in extract, I mean isomerase or conjugate. It seems to be a very time-consuming job to isolate. Probably due to that tough isolation work, the price is quite expensive. Irimaquinone belongs to methylcesspepinol. This one disrupts membrane, but there seems to be more biological effect. Isolation is easy. Latrantine A is a macrolide and it binds to actin and has been used for many years. The source sponge distributes from widely from Okinawa, Indonesia, down to Vanuatu, Fiji, at least. You can find this sponge a lot on the underwater wall of Bunaken National Park at Manado, but sporadically at other places. In addition to these molecules, there are more compounds. So you can supply some bioactive molecules to researchers worldwide, and also it may be beneficial to some local researchers. But you need good isolation work together with instruments to confirm the identity and purity. By the way, probably most of the audience are not familiar with sponging. So if you do have an interest, please attend the invited lecture given by Dr. Nicole DeVoe early this afternoon, later this afternoon. The latter part of the second section is on the same or similar molecule from different source organisms. Manganese A is a unique alkaloid showing anti-malarial activity. Originally, it was discovered by my former colleague, late Professor Higa, and they coined the name after the collection site Manza in Okinawa. They reported this molecule from two sponges. The above sponge seemed to be quite endemic to Okinawa area. But the other sponge, Arkansas Strongylophora indians, is quite widespread. And I saw this Arkansas Strongylophora sponge in Indonesia at many dive sites as shown in these pictures. From Savan to Sorong, and also some down to South Kapa. Some of them do not look the same, but judging from the internal texture or morphology, I believe that they are the same. The chemical contents may be different between colonies. Actually, Professor Tsukamoto of Kumamoto University in Japan and her collaborators at INSURA reported some unique analogs as shown right. Another example is the two sponges. They look quite different, but containing the same molecule but different way. Particularly, Cacosporidium microphysiciensis contain two more molecules named lorimalide and xamphanolide, both of which target microtubules. Therefore, biosynthesis genes are of interest by several research groups. However, it was not reported yet. As we can access both sponges in Okinawa, I supplied 3 specimens each to my collaborator to examine the genetic content. They found that there seemed to be not so much difference on the bacterial fauna for each species, but quite different between species. Also, we expect that the bacterial response for producing methylene can be found easily, maybe through we expected that horizontal gene transfer or same bacteria may live inside, but it was not that easy. Other one is rheumatism. In addition to the previous sponge on the left, different shaped sponge also contain the same molecule, but we have not worked on the right sponge at all. For halcomramide, there is a very similar looking Thai sponge containing the same class of macrolide, namely cabiramide. Also, cabiramide was found from originally from Okinawan migrants. We have not identified which sponge contains the molecule. So, we have not examined the genome of cabiramide sponge here. Cellular sponges. One of the most studied sponges in marine natural flora contain similar compound, namely misakinolide and sphenoide, with or without double bond at this position. Although the outer morphology is a little different, but internal texture are nearly the same. The genes for misakinolide was already sequenced years ago, but one for sphenoide was not reported yet. Although in the literature, cyanobacterial sphenoide was already reported. I heard that the same group worked for misakinolide, also worked on this sphenoide, but so far I have not seen their paper on sphenoide. Anyhow, on the origin of the unique metabolite, there has been a lot of discussion what kind of microbial sources or not. But there are only a handful of results on the biosynthesis genes of sponge metabolite. Summarize the second and the last part of my talk. We can supply molecules for life science studies from an orange. But there are a lot of unknowns how the molecules are produced in these organs. And the metabolite variation exists even within the same species. Okay. The last part of my talk is on the chemical and genetic diversity of two groups of organisms. First on the soft flora of the genus Phyton, and second on the Pilidia nudibranch. Soft flora at shallow waters in coral reefs are easy targets for natural product studies from early stage of marine natural products. And a lot of molecules, mainly diterpenoids and sesquiterpenoids, have been reported. Soft corals of the genus Phyton shaped like mushrooms, and they are rich in membranoid class metabolite, as shown here. About 30 years ago, I was asked to check one of the molecules named Phyto-A as a source in Okinawa, because this molecule was reported to have anti-tumor promoting activity, and someone wanted to develop it. also they quickly soon after we started. And we did a chemical screening on the soft soil. As a result, the most dominant species was found to be consist of many chemicals shown here. Then we examined further on this group of organs with 3 viewpoints: chemical, morphological, and genetic diversity, together with biologists. As a result, one species, Sulfite Tokyoform, collected out of Okinawa Island, is quite uniform in the 3 viewpoints. But the most abundant species, Sulfite glaucum, was found to be quite complex, and it is likely to be consist of several clades or species discussed by biologist Professor McFarlane in the United States. At that time, she already reported the species were divided into 6 clades. And more recently, Maloney in the United States and also LC, the collaborator of McFarlane, and our collaborators, including us, reported the chemical and molecular diversity of Parawan sulfite by analyzing mass spectral data of pure compounds and its rock with a platform named GNPS, Global Natural Products Social Molecular Networks, which is available to anyone. As a result, the species sulfite glaucum is consisted of mainly 2 clades: blue clade B and orange clade F, in addition to another species, sulfite Tokyoform. With GNPS works, the chemicals can be grouped into the right figure. And each circle represents molecule or related ions. And we may understand that some compounds can be found more in color blue or clade B, and the other in orange or clade F. It is not so clear, but it shows the complex nature of the species. Second group of organs we have interest are nudibranchs, cell-less mollusks, or sea slugs. When we worked on a few species as shown in this slide, we may understand that each species developed sequester unique chemicals for their own details, and they developed it during evolution. And we looked into the most dominant species, most abundant species of the era in Okinawa. By collecting only 16 specimens, we examined their dominant chemicals and molecular phylogeny. As a result, one of the molecule, compound 1, was found from 5 specimens with the same sequence. So we expected that it might be the beginning of the differentiation of the species. To confirm this phenomena, we collected more specimens. After analyzing molecular phylogeny, the species were grouped into 5 species and 6 clades. And with chemical analysis, each clade may express a unique combination of molecules by showing the compound as numbers. But it is not easy to understand how they are complex with this figure. So my student also used the GNPS to analyze the diversity. As a result, the molecules are separated roughly into 3 groups. One is Becquerel type bicyclic compound. Second one is monocyclic or other class of cyclic molecules, and one linear compound. And coloration of each circle indicates a clade. And you can see some compounds can be found only in a few clades. All right, but all for my talks, and I like to summarize. Indonesia has apparently has wide environment, and it's various places and explored places. So, and also as everybody knows, it is located at the center of biodiversity and for triangle. So there must be a lot of bioresources in your water waiting for your research. And for marine nature product industry, it has passed at least more than 5 decades. But there still gives a lot of chances for discovering not only new molecules, but new biological phenomena. And of course, everybody needs motivation to work on each field. It is, in my case, it is fun to observe unique marine organisms in the field and also discover new fields in the lab. Okay. Thank you for your attention. That's all my talk.
Okay, thank you very much, Professor Tanaka, for interesting talk. And now we are moving to our next speaker, Tanaka Sensei. Hope you don't mind to wait before Q&A session. Thank you.
Yes.
Okay, our second presenter is Bapak Insinyur Mastur MSc PhD. Currently, Bapak Mastur is a president of Indonesian Biotechnology Consortium and head of research center for biotechnology and agricultural genetic resources. It's under Agency for Agricultural Research and Development, Ministry of Agriculture. He achieved his doctoral degree on bioregulation, Tokyo University in 1999, while his master and bachelor degree was from IPB University, Indonesia, in soil science and conservation, which is obtained in 1994 and 1988, respectively. Bapak Mastur will present in a video form, and please welcome Pak Mastur via video, who will be presenting the current research and biotechnology to enhance crop improvement in Indonesia. Okay, time is for Pak Mastur video.
Everybody. Today I will talk about the current research on biotechnology to enhance crop improvement in Indonesia. Agriculture sector is an important sector in national agricultural development in Indonesia. The sector is very strategic related to food security and agriculture-based economic development. Agricultural development requires support system from human resources, science and technology, genetic, land and water resources, agricultural input and infrastructure, and proper financial and policy system. In this term, agricultural development is directed to achieve advanced, self-sufficient, and modern agriculture. Agricultural development targeted to increase farm production and productivity, land area, and its supported system, production efficiency, which is supported by research, innovation, and mechanization. Indonesia has got target to achieve self-sufficient in several strategic commodities such as rice, maize, soybean, sugarcane, chili, salad, garlic, and cattle toward Indonesia feed the world by 2045. The ambitious target is important and required to set focus and synergy within resources to achieve the goal. However, there are several challenges in future agriculture, including expensive land and labor costs, water scarcity, and expensive costs, decrease on productive land for agricultural practices that force farming system on marginal and suboptimal land, and challenges on climate change impact on biotic and abiotic stress. On the other hand, high demand on healthy food lifestyle and environmental friendly also challenges people who work on agricultural biotechnology to develop crop varieties to meet this demand and challenges. Diversity of plant genetic resources, or PGR, is fundamental resource to develop a new plant variety or crop improvement. There are two basic conditions of agricultural practices that determine sustainable or not sustainable plant genetic resources in the future. At diverse GPGR with normal income condition, development and commercialization of particular local superior variety without conservation activities in the region will lead to non-sustainable agricultural practices that will create biotic and abiotic problems in the future. On the other hand, involvement of in situ conservation by maintaining variation of local varieties in the area will bring sustainable production and commercialization of new varieties by crop improvement in the future. There are several in situ conservation methods that exist in forestry, for example, sagu plantation in forest area, or farming practices in arable area. The other method as a backup in plant conservation in ex situ collection, which applied in the field collection or in the seed bank. Indonesia has several institutes managing ex situ gene bank collection, both in the field and seed bank. ISIBIO collecting thousand accession of food crops, for example, rice, maize, soybean, sorghum, mung bean, peanuts, and other edible beans, root and tuber crop. In ISIBIO, we also have microbes collection and insect specimen. Other research institutes will gene bank collection, for example, some Indonesian Research Institute for rice, tuber, and beans, ornamental crop, vegetable, and fruit that cover about 70,000 accession, plantation crops about 2,000 in total. As my previous statement, there is biodiversity is important for crop improvement in the future. Fast development on biotechnology enables new techniques to develop new variety. Conference breeding requires long-time crop and enable molecular marker enhanced development of new varieties by molecular marker selection and new breeding techniques with application of genetic engineering by transgenic and genome editing technology. We fit in compatibility on intra or interspecies to create either what we call genetic modified organism or GMO and also non-GMO products. Other progress on in vitro study highlights inter-endosperm culture, protoplast fusion, and in vitro mutation on development of new varieties. Now I will start the topic on in vitro technology. There are three inventions that open pathway for development of in vitro technology. We can say that first invention to cell totipotency-based Swan and Sladen in 1838 was a pioneer for in vitro technology, followed by Harberland in 1902, who culture isolated cell and invention in vitro germination media by Knudsen in 1921. The current research in vitro technology, especially in ISIBIO, we develop micropropagation techniques for many crops, for instance, in root and tuber crops, horticultural crop, and industrial crop, which is some of them oil palm, sugarcane, bamboo, and some medical plants. Development of new improved superior variety has been released, for instance, on sorghum, rice, citrus, and chili. Application of in vitro technique, for example, in rice breeding, we utilize application of anti-culture to enable development of superior line from double haploid breeds and combination of in vitro mutagenesis and selection to obtain new rice varieties for saline condition. Some new improved varieties using bioprima, bioemas, biosalin and 1 and biosalin 2. Next, I will talk about the sequencing and marker assessment for breeding and crop improvement. PGR including modern cultivar, breeding line, local variety, land races, and crop relative species are very important as input into selection process through plant breeding to develop new and improved varieties. Out of species, small of high cultivated plant species were identified and fruit crops are at loss proportion. Therefore, it is important to set wise sustainable utilization of PGR to achieve food security using advanced technology, especially genomics. The genomic sequencing basically use DNA and also accompanied by RNA for gene expression that could specify a particular protein. All generated data from genome sequencing and further analysis will be for biological sequence of genome test, and importantly, they are valuable and applicable further analysis. Where do we do genomic sequencing? With this technology, scientists are able to read genetic information. There are three types of genomic. First is comparative genomic. The second, functional genomic, involved in study on functioning and regulation of gene expression and metabolic pathway. And the third is structural genomic, which is determine complete sequence and gene map. Here are some examples of the application of genomics using survival on crop improvement, such as genomic size, gene number, gene mapping and sequencing, evolution of crop plants, gene cloning, identification of DNA marker that can be developed as marker selection, transgenic breeding, linkage map, and ETL mapping. Importantly, this genome sequencing allows scientists for better prediction and phenotypic character on particular genotype called genome-based breeding. This improves and new varieties and related technology could be beneficial not only for human healthy, also for food safety, but also better environment. One important achievement is establishment of pertanian Indonesia, as can be accessed in this link, www.litbangotpertanian.go.id, but in copyright in 2018. Genomic information of 11 species is featured the PGBA based on genomic sequencing and genetic mapping. Users are able to look genome browser of some crop with detailed information in genome. This PGBA provides variant for marker useful for DNA fingerprinting analysis and important source of molecular marker and gene governing design traits. Marker assisted selection is main tool for molecular breeding. Especially genetic marker are linked to genetic variant affecting phenotypic change. Second process using molecular marker including single locus or genomic-based marker could be done in seeding phase, so it will reduce phenotypic selection and breeding marker as MAS to assist breeding. In case of marker-assisted background selection, this could be high-density molecular to detect QTL. ELJ also developed marker kit to detect early maturity of sugar palm that access breeding and selection of local accession. In addition, several new varieties have been released and ongoing using marker-assisted selection estimated by Impari HDP, Impari Plus, Impari 40, Biopatenggang, and as essential variety. Marker-assisted backcross selection have been used for developing Bioni sestigera, which is resistant to brown plant hopper. Molecular breeding also surprise with total spiglet number character higher in biofortification of iron content on rice. And last technique, I will discuss this genetic engineering, transgenic and genome editing. Transgenic, also known as recombinant DNA technology or gene manipulation, that alter gene in living organism to produce genetic modified organism with a new genotype. Various kind of modification are possible inside a foreign gene from one species into another species, genus or higher taxa, altering the existing gene to change the gene expression. Thus change genotype and phenotype, new product or variety. The genetic modification able to overcome sexual barrier incompatibility, and genetic engineering technology can be applied to improve product and environmental friendly. There are three-step genetic engineering research. First, gene cloning, where we can clone gene from unlimited gene source, including from different species, genera, or kingdom. This selected gene modified by molecular by addition of promoter and terminator to be constructed in expression factor. The second is gene transfer to introduce target gene to direct transfer was applied with bombardment particle carbide silica. And Indirect transfer was done by agrobacterium to Mifasian vector. The last step component is gene expression, where targeted gene cell be expressed and change the intended phenotypic character. There are several national institutions working on genetic engineering to produce GMO. For example, ISIBIO has been developed Biogranola agriorti, GMO potato resistant to Phytophthora infestan. Developed by collaboration ISIBIO and Cornell University. Also, drought research sugarcane already released by PTPN9. Other research are developed by insertion of alanine aminotransferase gene driven by root-specific promoter OSIRE. And the current status of this study in confined field trial to select baseline. There's also tomato GMO for CMP and TYLCV virus resistant, which is currently on multiple trial CVT for food and environmental safety. The next technology is genome editing. This technology modifies gene or genome by inserting, replacing, deleting, and transferring precisely with the help to an endonuclease Cas9 enzyme as molecular scissor on specific target. Some example genome editing system are GFN, Talent, and CRISPR-Cas9. CRISPR-Cas9 currently is most widely used and preferred because the system is more effective and editing application, drug discovery, and gene therapy to cure genetic disease. A pipeline for using CRISPR-Cas9 mediated genome editing to generate plant knockout mutant started by CRISPR-Cas9 construction and interrogated plant or known as plant transformation. Screening and regeneration of selected transformation were assessed, followed by genetic and phenotypic analysis on transformation plant. Genome editing system has been concerned in some university and research institute in Indonesia, not only ISIBIO, but also Gajah Mada University, Bandung Technology Institute, and also Indonesia Research Institute for Biotechnology and Bioengineering. ISIBIO already worked on a CRISPR-Cas9 genome editing system since 2016. Research objective to obtain rice improved variety for semi-dwarf character and potassium efficiency. There are also CPVD virus-resistant citrus, increasing artemisinin content and chili resistant to Gemini virus. The CRISPR-Cas9 genome editing research from Gajah to get albino or seed by mutation BDS3 achievement. Ministry of Agriculture, University such as ISIBIO, UGM, UNED, and research institutes such as LIPI also have been conducting research in biotechnology. Indonesia Institute of Science also obtained some new crop variety, either from soma clonal variation, marker resistant, irradiation, or genetic engineering technology. Biotechnology has contributed a lot on crops improvement and new varieties released to meet national program or human need and environmental friendly in general. ISIBIO has been released some improved varieties. From example, from marker selection, we released 5 rice and 1 chili improved variety. In vitro technology become a great tool to produce 4 rice, 1 chili, 1 tangerine, and 3 sorghum new improved varieties. Regarding modern biotechnology research, Indonesia has released already drought-resistant sugarcane by UNED and Phytophthora infestan resistant potato by ISIBIO. And from latest technology, CRISPR-Cas9 genome. That's all my presentation. Thank you very much.
Thank you, Pak Mastur, for a nice video talk. Okay, ladies and gentlemen, question for Bapak Mastur will be written in chat, and the committee will send your question directly to Dr. Mastur by an email, and I'm sorry about this. You can wait for the answer after the committee sent an email to Dr. Mastur. Okay, so now we back to Professor Tanaka. Tanaka Sensei, are you there still?
Yes, I am.
Okay, to remind us on the lecture from Professor Tanaka, it was very, very interesting, Sensei. There were three topics actually, and interesting one is the Professor Tanaka closing with remark that it is a big chance still for Indonesia to find new compound in the lab due to the unique location of Indonesia marine. Okay, so in chat we have 4 questions, Sensei.
Yes.
I will read. I will read them. Yes, the first one is from Ibu Yakti, Pusat Studi Biotechnology UGM. I just wondering how you realize that you identified the new structure that have a potential to be developed in the future. Okay, maybe one by one, Sensei.
Okay.
Or...
Yeah, of course. I just replied short on the message, but I'm going to answer directly. Of course, when you want to develop the molecule into some other application or something, in that case, the original molecule is expected to have some biological activity usually. In case of, for example, halichondramide, it targets G-actin in our cells, and or just showing some toxic activity or antifungal activity. But in addition to that, it might be useful, for example, by attaching some kind of antibody. There is so-called drug group named antibody drug conjugate in ADC in short. So because of that, there is some easy to attaching point in the molecule structure. It might be useful if the molecule can be supplied in a synthetic biology method or recombinant biotechnology technique. But it's not it's a totally different field from my own. But it's not easy to supply. We better not think that we supply the molecule from nature, but by technology technique. Is that all right?
Yeah. Okay. I forward to number 2. It's from Ibu Tuti Murniasih. It's in Ancol. I think you visited there already.
Maybe Tuti is the one I met already many years ago.
Okay. Yes. His question is. For compounds that have been applied as life science reagent, it is produced in synthesis process. Is there any most effective strategy for the selection of compounds from the isolation until applied?
Okay.
Sensei, we will have time until 11:28, and we still have 3 questions. So please, be short and Okay.
Let me do. One example is that so-called domoic acid or kainic acid. Once a simple molecule like kainic acid it has been supplied from nature. But nowadays, I believe some synthesis people made a strategy, so it is supplied by synthetically in part. And also, but in the actual drug, like so-called eribulin, it is against breast cancer, and this drug is quite complex in structure. But since the people also produce the molecule, because the in this case medicine is quite expensive, so you can it will be there is a reward for supplying by synthetically. So it is not simply on the structure, just by the structure, but the values of the molecule.
Okay.
Is that all right?
Can I read the third one?
Yep.
Yes. Okay. It's the question is from Ibu Tika. It's a researcher from BRIN. Thank you, Professor Tanaka, for the nice and interesting presentation. I have a question with regards to the diversity of marine secondary metabolites or bioactive compound. Are there any influence on specificity in the taxonomy, in the species or family of respected organism, or do you think it was rather affected more by geographical or environmental factors or microbial symbiont? Thank you.
Okay. Judging from the molecular diversity, microbial resources, and also some among the cells-time organs, sponges are the most prolific sources of various molecules. In case of, for example, corals, soft corals, and also stony corals, they are not. They provide a huge number of terpenoids, but from the structural diversity, they are not really exciting. Okay, and what is it? Geographic and factors. Yeah, it's not that easy. We cannot answer easily. There are many cases, and in some, it was already known that some organs are quite different between colonies. And in other case, most any colony contains almost the same molecule. So, and there are, of course, some people examine microbiomes in bio, but it's not easy to conclude as a fauna, a microbial fauna in this kind of organism. As you can see in some of the literature, after obtaining so-called metagenomic DNA from sponges, you can find many groups of bacteria.
Okay, the last one is from Pak ISIBIO. It's about modification of the structure, the functional groups, or modifying the elongation of carbon bond. Are there any general rule or about steps that we have to consider first for modifying the compound structure? And the second one is a huge marine biodiversity exhibits a huge diversity of marine-based biochemical as well. So what is the current strategy in order to accelerate the discovery and development of drugs from marine organisms? So,
for the first question, in case of for some derivatives targeting protein, because of the huge size of the protein of the compared from the so-called ligand molecule, like natural product, probably we may need some we may need to attach some linker moiety, but I don't have much experience on this aspect, and it is better to consult other research papers when they target the target protein with the natural product. And for the second question.
Yeah, for the second question.
Yeah. Nowadays, big pharmaceutical companies do not prepare a natural product library by themselves, but small venture-like companies prepare. What I heard is we can provide library of natural products, and these companies screen against for some drug target. And find out the hit, I believe.
Okay, that's all, I think. If you have any question more, I think can we write an email to you, Prof. Tanaka?
What?
If the audience have any more questions, because we have a limited with the time. So can we send an email to you?
Oh, yeah, no problem.
Okay, thank you very much. Okay, thank you very much for Professor Tanaka and Dr. Mastur for addressing very interesting topics. I won't take a summary because it's very, very deep and well, I think it's very, very broad, also an interesting topic, and I think a lot of our researchers here is eager to actually make communication with you. Yeah. Okay, on behalf of the committee, I would like to thank to all speakers and to participants to the active participation. Thank you for joining us today, Sensei, and also for all of you participants. Now I'm giving back the time to the MC. Yeah, I think the time is up and exactly, okay, thank you, Mbak Wibit. Thank you, Professor Tanaka. And
thank you, you are welcome.
Giving applause, big applause to the. Thank you.
Thank you.
Thank you, Professor Ecology, and also Professor Tanaka and Dr. Mastur for a very insightful session. Distinguished guests, ladies and gentlemen, we would like to remind you to please kindly fill out the attendance list of the first session that has been informed in the chat feature. Now, for the next agenda is Principle and Mechanism of Biotechnology Product Session. We will hear presentation from Mr. Alfian Mauludin from Ditek Jaya. Mr. Alfian Mauludin, you would have 30 minutes and the time is yours.
Okay, thank you. Let me share my presentation first.
Sure.
Is it already up here?
Yes, it is up here already.
Okay.
The time is yours, Mr. Alfian.
Thank you. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. I'm very pleased to see you all. Thank you, and thank you for coming to this presentation. And really thank you to for give me this kindness of opportunity so I can be hand in here. Before I start my presentation and our discussion in this opportunity, let me explain to you and give the company profile for Detect Jaya. Maybe I share first.
Excuse me, Mr. Alfian, the sound, we
Sorry?
cannot hear the sound. Maybe.
Oh yeah, sorry. Sorry. I will
the sound with the video.
share the sound. Okay. So.
Okay.
Okay, thank you for your attention to see my company profile video. Before that, allow me to introduce myself. My name is Alfian Mauludin. I'm from sales engineer PT Detect Jaya. As you know before, PT Detect Jaya as a solo agent Shimadzu Indonesia. Okay, in this opportunity, I will share to you all about the analytical intelligence with Shimadzu solution. What is the analytical intelligence and what the Shimadzu solution for facing the next future maybe inquiry should be in the next future. Okay. Like we know, there are some difficulties in operation, such as diversification of working. Right? There are two types of diversification of working. The first is work efficiency, and then the second is variety user. Okay, as we know, day by day, the challenges in work are always increased, right? Such as the challenges for the increased job, many job or many tasks with a limited time. So what we need in here is how the instrument, how the technology can be support us to give them good efficiency or operator efficiency, right? And then for the variety user, there are two types. Skill operator, is it like export operator? Okay, maybe no problem or no issue for the skilled operator or skill export operator, but how the beginner? For the beginner, maybe need the more technology or more help to that the task for this. How make everyone, even beginner user, can use it the instrument as easy as they can. Okay. Then What the Shimatsu want to introduce to you all is it the analytical intelligence. Analytical intelligence in here means a smart analytical platform that incorporates state-of-the-art IoT. Is it like Internet of Things, or everything about internet, everything about networking in here, and then analytical intelligence and machine learning algorithm functionality. So is it like machine we test machine to learning the algorithm function so that the machine can helpful to make our job, our task in laboratory especially to be easy. Okay, and then the analytical intelligence means to automation workflow include auto monitoring, auto controlling, auto diagnostic, and auto recovery function release the user from routine instrument operators. So when instrument have problem, instrument can be what I can say can be recovery without our help, something like that. And then what the purpose for analytical intelligence? Is it like for minimizing system downtime and then optimizing efficiency? When we get the minimizing system downtime and optimizing efficiency, so the productivity, our productivity will be increased. We will get the maximum productivity in our activity, especially in laboratory. Okay, next. What instrument have implemented this analytical intelligence? Okay, the first of thing is ultra high performance liquid chromatograph. Is it like UHPLC, right? And then as you see in my slide, in my presentation, is it the newest UHPLC from Shimatsu? We call it next era series. It means like next era or next generation era for facing any diversification of the working. Okay, this is type is it actually for modular system, so very flexibility for upgradable and as your own configuration and as your need in the laboratory. Okay, then. So what the intelligence in our HPLC, maybe you can see in here, this is like a routine workflow, which we always do in the laboratory, right? The first time we start up and then we do set up mobile phase. Is it like a important thing when we're using chromatography, and then we do column equilibrium, or we waiting until the system stability. So after system stabilize, we do analysis, start analysis. We inject a sample or standard in there. When the system running, we need to check, is it mobile phase check or is it enough or not? After that, we get a result and then we do system monitoring and a shutdown. And then we will using the instrument in next project or in another day. Okay, so how about the intelligent workflow? Actually, the intelligent workflow, like I showing in here, we can see every manual process can be run as automatically processed, such as auto startup. We can set instrument and accordingly what time we set the instrument will be turned on even we not yet arrive at the laboratory. Is it possible to do that? And then do mobile phase setup. So for mobile phase setup in here, we have a technology intelligence in here. We call it advanced real-time mobile phase monitoring. What is the advanced real-time mobile phase monitoring? Is it actually this feature for mobile phase setup? We have real-time mobile phase monitoring in here. We know the important things in HPLC. Some of them is mobile phase, right? When we not care about the capacity of mobile phase, it can be problem for our instrument and for our analysis. Actually, in here for mobile phase monitoring, is it like a sensor? Maybe I will show you in here. This is the sensor in here, gravity sensor. So when you put the mobile phase bottle in here, contain mobile phase sure in here. So the system will be calculate automatically can be calculated automatically how much volume inside the bottle, right? Then we can set up the minimum volume for the mobile phase in here. So when the system decrease until the minimum volume which already we set, this it will be send the notification in case of shortage. So the mobile phase time monitoring can be installed and can be directly using in our smart gadget, our smart device, like maybe smartphone or something like that. And then what the purpose for advanced real-time mobile phase monitoring? Sure, is it for prevent system dry up? Is it like when we using the instrumentation and the mobile phase to be dry up? Is it will be impact to our column? So column will be easy to damage, and then another that the result or the analysis will be not good as well. So we need to reanalysis, so wasting time for this. So real mobile phase monitoring, is it means maximum of time. Okay, this is for one patent from Shimadzu for analytical intelligence. Is it advanced real-time mobile phase monitoring? Okay, then the next, after we make sure for mobile phase, then we do monitoring or column equilibrium in here until the column stable, until the system stable before we inject a sample or standard. For the conventional method, we do conditioning column with plusing instrument with set up flow rate, example like this. Maybe we set up maybe 1 milli per minute or more, something like that. When we first turn on the instrument, turn on the pump, will be make a pressure jump in here. If you using the higher flow rate, right, and then pressure jump in here will be impact to your lifetime column. Even that, so Shimadzu released analytical intelligence technology for flow pilot. Actually, flow pilot is it around the mobile phase flow rate gradually to set the point automatically. Is it mean when we set up 1 milli per minute in here, the system automatic run in a half of flow rate? Is it like 0.5 milli per minute in here, the example, and then when the temperature increase gradually, the flow rate will increase too. So this is prevent the extra pressure, so will be save your column lifetime. Okay. Another thing from the flow pilot protect column, is it everything automatically? You just set up in the software, and the software will be start your method, and then running the flow pilot protect column technology here. Okay. Then after we get the system stabilized, and then the column equilibrium, we got the good stabilization in there, we starting to start analysis, right? And then when we starting the analysis, we need to check the mobile phase, is it okay or not, enough or not, something like that, and then do system check. Maybe sometimes we get no good result when we do analysis. Is it can be because is it because maybe from the instrument, or maybe from our sample itself? Okay, so in the Shimadzu, they have another analytical intelligence. We call it auto diagnostic and auto recovery. It's mean like instrument can diagnostic if there are something problem in the instrument, but they can recovery too by themselves. Okay, so no need our help to recovery the instrument. Okay, what's the meaning in this slide is like this. When this normal pressure, right, when we do analysis in here, we inject, okay, in normal pressure, we will get the good result in here. But when sometimes the instrument or the pump, there are a bubble maybe in the mobile phase, so the pressure will jump the pressure will drop. So make a fluctuation in here. Pressure will be fluctuation. So when we inject in this condition, you will get no good result from your analysis, because that after we on this feature auto diagnostic, instrument will do auto purge to prevent this abnormal pressure. After auto purge, and automatically system will give the normal pressure to normal pressure, and then conditioning again. After everything okay, system automatically will be re-inject your sample. So if the failure in the sample too, and then auto purge happen in here, the system will be re-inject sample to before. So this data will destroy and not saved in the database. So you will get the great result without you must waiting in the laboratory. So the benefit from this, yes, the chemical does not need to be physically present when abnormal condition appears. So after you preparation, after you start injection, you can go out, you can do another job, another task. Then you will get the result even in there. Next series can perform auto correction, ensure maximum system uptimes. Okay. Okay, and then after we do auto diagnostic and recovery, we got the result, and then we are reporting. So we calculate the result. We need to make sure if our instrument have a good maintenance schedule. So the Symposium solution give a smart service net. It means like some feature in our software, some feature in our chromatography software, like give the approximate maintenance timing by prediction from the use history. It's mean like when you must calibrate, when you must replace another part of like a consumable part or even spare part in there. You will not missing for the maintenance your instrument. If your instrument highly good maintenance, I think the lifetime is good to write, and then your result will be great. Okay. Is it that's all the simplest and intelligent workflow from Shimatsu? Is it our intelligent workflow automatically intelligent workflow in here? So important and so helpful for us in the next project. Okay. And the next slide, I want to introduce to you the newest gas chromatography. We're already talking about liquid chromatography, right? Okay, so I want to share with you all about the gas chromatography. We call it next industrial standard GC generation. We introduce to you about a new experience, intuitive and simple controller like a touchscreen GUI, graphic user interface, and then what the meaning graphic user interface is mean like what the display in the GC instrument is it similar or same in the software too. So easy to make so easy to use this GC. And then we have new graphical icon improve operability. It's mean like the easy friendly use for your laboratory activity. And then we have innovative software enhancement like quick batch. Quick batch mean like when you are watching a movie in studio, the software will info you what the number via empty are already fill up, and then you cannot fill again if already taken by another file. Like similar like that. And then we have lab solution direct. Is it enable remote control and monitoring? Yeah, is it really useful when you're using when you working like a work from home? You can direct access. You can connect to your smart gadget, something like a smartphone or tablet. Is it another tools can be connected in there? And then the important thing in our new GC is tool-free routine maintenance. like I introduced you before, there are some talking about variety user rate. There are two type user. Is it a beginner and expert? This new analytical intelligence from Shimatsu purpose will help the beginner user to make everything easy. First, we need click tech nut. Is it like for replacing like a septum or glass insert? Maybe you can see in this tutorial video to make it easy to be understanding. There is just only need 30 seconds to replace stone or glass insert. We can compare with a previous technology. We need tools in there, so need time to replace it. So you can so the instrument another benefit is like save your time. And then how about the click tech connector in here? Click tech connector in here means when you install new column, so you don't need anything tools be used in here. Just only click tech connector, just open the GC and then you plug and play to the injector port. Something like this, so easy to beginner user. Like this, and then we have a smart jig. Smart jig, it's mean like a jig for make sure everything already according a GC requirement. Depend the detector you use it and the type of column you use it in the gas chromatography. So you need jig to measure how long the column installed to the injector port or to the column port, to the detector port. And then the simplest thing in new GC Shimatsu technology is it like a breeder oven. Breeder oven, maybe some people know notice for the lights we need inside the GC to get us when we do installation or when we do maintenance in the GC. Okay, Shimatsu already have a breeder oven inside the GC column oven. We have to clean gas flow line in here. The point is we just need a plug and play the filter when we do installation or when we replace a new filter in here. Okay, as you see in my presentation, you freely create your own configuration. Shimatsu give the flexibility for your configuration. You want to you want to create your own configuration, like such as you want to add with headspace system auto sampler, or you want to connect with thermal desorption or pyrolyzer or something like that. And actually, some of this configuration already installed in Brain. Or ISIBIO or another city. So in this slide, let me introduce to you all another product from Detect.jr products. We can over to you. We can give your solution for your inquiry in the laboratory activity, maybe in the simplest like a spectrophotometer. We have a molecular spectrophotometer, UVVs, even like from the double beam spectrophotometer until a single beam spectrophotometer. And then we have a Fourier transfer infrared, or we call it FTIR. You can see in here like a portable FTIR. You can move the FTIR to another place. Is it ER Spirit? And then we have a Huygens ER. Is it ER tracer can be upgraded until near. Okay, then we have spectrophotometer atomic. We have ACE. ACE is from like elemental analysis, tracer analysis. You can use it to measure elemental analysis from percent and up to the ppm. And then we have ICP and an ICP-MS. Is it more sensitivity for spectrophotometer atomic, which can measure until PPT levels. And then we just we have a chromatography, HPLC and GC, and then we have mass spectrophotometry such as like LCMS, single quad or triple quad, LCMS MS, and then we have GCMS single quad and GCMS triple quad too. And not forget, we have another solution for your unknown analysis. Is it your QTOF MS? MS from Shimatsu, which can combine with imaging imaging system IMScope. Is it in the process installation in LIPI? And then we have another product like thermal analyzer, like differential scanning colorimeter, and then TGA. We have another solution like for MALDI. Is it already have in LIPI2 in brain performance MALDI TOF in there. So we have a newest MALDI like a MALDI Mini and MALDI 8020 for the benchtop MALDI. We have another instrument like total organic compound for your environmental analysis. Is it like a simple instrument? Another simple instrument like balance, analytical balance, or top loading. Okay. The next is it like I want to introduce to you all. Beside the instrumentation, we have another solution for your laboratory environment. Is it we call it a software solution? The challenges from this inquiry from every single laboratory is how to integrate the process with instrument become one solution. Is it we have a solution using a software. We have lab solution CS or client server like you see in here. We have lab solution CS which can connect more instrumentation in here. Even like not only chromatography system, we can connect a spectrophotometer system, and even we can connect another brands. So we under one platform software, we call it lab solution. So you can controller, you can reporting, you can do analysis in everywhere you want. As well, it in the same domain, in the same network. Okay. Like you see before, is it the newest digital transformation for your laboratory? Because another product, even like lab solution CS, we have a LIMS laboratory instrument management system. We have QMS, we have DMS, and Nicole training. So is it a full package for your digital transformation of your laboratory? Okay, this is the digital technology has been growing rapidly. So maybe we know we cannot notice if we need the technology as like analytical intelligence for our activity. Okay,
Excuse me, Mr. Alfian, you will have 3 minutes left. Thank you.
Okay, this is actually the last slide from
Okay, that's great.
Okay, so because we don't have much time in here, please, I want to make sure I want to point to you if you have another inquiry. Is it like not only you need an instrument, if you need the method or you need to maintenance or service or something like that, please don't hesitate to call me, to call my team, to call my company, to info our customer service in here. You can save my contact number in here, and you can reach my email in here in alfian@detectjaya.co.id or sales@detectjaya.co.id, and you can visit to our website. Is it detectjaya.co.id. Please don't hesitate to contact us in here. Okay, thank you, and see you, and nice to see you all in the great webinar today. Thank you. I will back to our moderator.
Thank you, Mr. Alfian Mauludin, for a great presentation. Ladies and gentlemen, before continuing to the next agenda, there would be some documentary videos from our partnership that we can watch. So let us watch the video together. Here we go.
Since 2019, PT Wadia Prima Mulia has continued to develop in meeting the needs of basic laboratories for Indonesian territories. Until now, our company has been trusted as an authorized distributor for 11 companies that produce the best laboratory equipment in the world. Our highly skilled and experienced sales team spread across Indonesia from Medan, Jakarta, Bandung, Semarang, and Surabaya. Our products. Sales services, including application and technical support, installation and training, also service and maintenance.
In the life sciences, researchers are actively pursuing studies aimed at understanding life, the causes of disease, and achieving breakthroughs in regenerative medicine. To observe and accurately measure intracellular processes and structures, researchers need a microscope that combines sensitivity and speed. Increasingly, scientists require the capability to view cells and tissue at high resolution, as well as their larger context with macro to micro capability. All of these advanced features must be easy to use so that even novice users can acquire high-quality data quickly and simply. Olympus created the FV3000 series to satisfy these needs. This next generation of confocal laser scanning microscopes combines leading-edge imaging features with renowned Olympus optics.
All right, the next video. The next video for the host. While waiting for the host, okay, here's the video.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porcine ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porcine pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah, tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik, tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porcine pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porcine pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3, yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche. Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol Start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat sampel mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porcine dari kami. Sekian dan terima kasih.
Now we will move on to the next video.
Halo, kali ini saya mau memperkenalkan alat laboratorium dari Syngene. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih, sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat, sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethidium bromida atau cybersafe DNA atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database tipe gel dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
And the last video.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
Alright, ladies and gentlemen, for the next, we will hear a presentation from the Director of PropLife Asia, Mrs. Soni Tababa. Mrs. Soni Tababa, the next 30 minutes is yours. You may proceed.
Thank you very much, Ibu Wiwit. Let me first share my screen and see if, are you able to see my screen now?
Yes, we are able to see it clearly.
Okay, let me just put that into slide mode. And can you hear me well?
Yes.
All right, thank you very much. So I'm standing, not really standing, I'm sitting right now, but I'm just the last speaker and your break is coming soon. But first of all, I'd like to thank the organizers of ISIBIO 8. This is a lot of work. Congratulations to BRIN and to KBI putting together all this conference, all the researchers with all this fantastic work they have done. And annually at that, since 2012, is really a lot of work. This is, CropLife Asia has a similar, I manage a similar program for 15 years annually, and it's a lot of work. Very much big congratulations. And the other one I'd like to say is that I just would like to thank you for our organizers and participants for allowing us, giving us a space to share our thoughts around plant science, around innovations, as well as around food security. And in the next few slides, before I go to the next few slides, so I am Ibu Soni, and right now I am in Philippines, in Los Baños, Laguna, but I work as a Biotechnology Affairs Director in CropLife Asia, which is based in Singapore. So the pandemic brought me another blessing to be with my family, spending more time with my family while working from home. So let me go next to my next topic, which is just to give you a flavor about CropLife Asia and its network. I always would like to introduce first CropLife Asia. We are a regional organization, and our global organization is CropLife International. CropLife Asia is based in Singapore. We have 15 national associations all the way from North Asia to Southeast Asia and down under. And you can see on your screen the 6 global R&D companies that are our members, and they are very much active in crop protection, seeds, and plant biotechnology space. And I said we have 50 national associations. You see them on screen, and I'm very happy that in Indonesia, we have CropLife Indonesia, which have been in Indonesia helping and contributing to societal goals in Indonesia is CropLife Indonesia, more than 3 decades there. And for our biotech program, you would notice that some of our national association focus mostly on crop protection. So for biotech and seeds, we partner with other organizations like in India, the Federation of Seed Industry of India, where they have a special interest group, which is Alliance for Agri-Innovation in India. We have the Council of Biotechnology Information in Japan, a CropLife international affiliate, and also China is so big that we have to kind of separate the program from a crop protection, purely crop protection, and for biotech. And as you see, we have 15 national associations here. Most of CropLife bring together the advocacies on not only crop protection, but plant biotech and seeds, but others just focus on crop protection. And CropLife associations, whether they are in the region or whether it is CropLife Indonesia, our vision is really to enable a productive food and agricultural systems through innovative crop science and technology. The vision is to have a living standards and improve living standards for not only our farmers, but for those who are using, who are involved in farming in a very sustainable way, and ultimately to improve and contribute to the important goal of food security. And so you would notice that our company members are really into developing innovations in crop science. So the major stakeholders where these technologies will be accessed are by our farmers. So we are hoping that our farmers will be able to grow more on less area through these innovative technologies, so that later on you could see that growing more in less area would help in our advocacies and our goals, overall societal goals in biodiversity conservation, and even in reducing the impact of climate change or in mitigating climate change. So just as a background here, CropLife, whether we talk of CropLife Indonesia, we have programs around crop protection, plant biotechnology, seeds, public affairs. Stewardship is very important. Stewardship is responsible care and anti-counterfeiting. And, you know, when it's a good product, it is likely to be copied. So we have to be very careful as well and make advocacy around anti-counterfeiting so that our farmers would have the guarantee that the products that they are using are genuine and would be able to benefit from it. So I'll just focus my next talk on global events in 2021. I think what I can say about 2021, there are so many big events that impact the way policy will be crafted in the next few, maybe in the short term, 3 years. But this will also impact us being scientists, or even from the industry. So let me just talk a little bit about COP15. So COP is a big police, but it stands for Conference of Parties. And this is about the Convention on Biological Diversity. So it is supposed to have been held in 2020 because this is held every 2 years. But because of the pandemic, this was started to be held in October this year. A high-level segment was started in China, in Kunming, and the theme is ecological civilization. So as you know, this is Convention on Biological Diversity, but this Convention on Biological Diversity is about conservation, about the use of biological diversity, and also benefit sharing. And so we have subsidiary agreements. So you must have heard about the Nagoya Protocol on the Cartagena Protocol first on biosafety, which is the only legal agreement around living modified organisms. You have the Nagoya Protocol on access and benefit sharing, and you have the Nagoya Kuala Lumpur Supplementary Protocol on liability and redress. So as you would note here in Kunming this October, last month, the high level, the parties to this called for urgent and integrated action to reflect biodiversity conservation in all sectors of the global economy. So whether you are in trade, so everybody is enjoined to really be accountable for biodiversity. So it's not limited to a few actors. And there's a call for increased investment and more private sector involvement. So there's a website here. This is where you can get updated news around what happened to COP15. This is a continuing, there's an ongoing negotiation that will be held in person next year in January in Montreal. So probably there will be some coming from Indonesia as party to the Cartagena Protocol to negotiate in this conference, in this convention. The other one I'd like to call is the UN Food System Summit. This one really focused on our food system so that we are called to deliver not only on the 17 Sustainable Development Goals, but really asking member states, the parties to this, to account, to make a reporting. And so, and make more solid commitments towards more sustainable, equitable food systems. So before the summit that was held in September, there were many independent summits and also summit by member states. In fact, as early as January, Indonesia held that summit around the Food System Summit. And so that is very important because maybe in the next 3 months or in beginning 2022, we will hear our leadership in different ministries to talk and set directions around our food systems, around our biodiversity. And the last one I talk is about, I will mention is around the Climate Change Conference, which recently happened in UK, in Glasgow. So this one really, they put together solid commitments. All the parties, they were asked to make their commitments instead of 2040 2040, move it earlier so that you can have global net zero emission. And the other point of this climate change conference is they also mention agriculture as making it more resilient to climate change. And if I just want to bring to your attention, just in case it has missed into your radar, that last month, the ASEAN adopted the framework for circular economy. And so here I put together the strategic priorities. It's around harmonization and mutual recognition. And we are talking now of circular products and services, around trade, around innovation, finance, so energy. And so these are, it encompasses a large sphere of the things that we are doing, and it will impact also the way research will be done and the directions of research that will be done, whether you are in the public sector or in the private sector. And we heard Vietnam is already, the Minister of Agriculture in Vietnam is already very interested in discussion about circular agriculture, and Thailand is also very interested. I have yet to hear from Indonesia how is the discussion around circular agriculture in particular. But it's very interesting. This is something that, everybody must be engaged. And so if we talk around, we go back, we remember Convention on Biological Diversity, Food System Summit, the climate change, this circular economy. I'll just highlight this common thread, not just the three of them, but for this talk, I'll just highlight the commonalities among those developments around the important role of agriculture, of innovations, technologies, and of partnerships. And this is so later on, I'll talk about that, but let me just segue a little bit and go back to the farmers, because whether it's climate change or whether we look at the food, every time we eat, we should thank the farmers. And because they are the frontliners, they are our heroes, they produce our food, but they are facing a lot of challenges, whether it be from climate, because climate could bring extreme weather, more rains or more severe drought, or more pests or changes in weeds and more diseases. And so these are a lot of challenges that farmers have to face, and we are also asking him, hey, produce more, produce sustainably. And so there's a lot of responsibilities that we bring on to the farmers, but we should also be in a position to be able to help the farmers, and that's why we're having this conference. So a lot of the disciplines that will be presented here will be able to find their way to farmers' hands. I'll just go back. This may be a 2014 research from the International Food Policy Research Institute, but this just highlights how important the contribution of technology in Indonesia. So whether they be in crop protection, they can increase yield as high as 20%, or in having crops that make use more efficiently of nitrogen, or having drought tolerant or salt tolerant crops. So when there is extreme weather as a result of this climate change, we see more the technology as a very important intervention to help mitigate or adapt in climate change. So this is another example, no-till. So for example, no-till help in not only in carbon sequestration, but it also double the maize. So if you don't till, you help in carbon sequestration, but it also help in improving yield. And this is the data, again, coming from the IFRI, which is a think tank research agency under the consultative group of international agricultural research. Now, just zeroing on some of the technologies in supplying food sustainably sustainably, particularly from a plant breeder's perspective, they can do a lot. They can develop varieties that can protect or reduce insect damage. They can develop novel herbicide tolerances or disease-resistant varieties, as mentioned with Master earlier, improvement of varieties that are more resistant to pests and diseases. And then we can also develop varieties that can, or farming systems that can improve carbon sequestration or improve tolerance to salinity, drought, or even improve the nutritional profile. And with the advances in science, we see, and we have been hearing, you are all scientists and researchers, so you have heard how novel molecular techniques and transgenics help in that direction. So Master earlier said that I'm very happy to hear that GM potato, earlier there was GM sugarcane, and now GM potato is now advancing, and maybe very soon we can see it really in the market after the variety registration approval of GM potato, and that is very good. And now we also heard from Master the gene editing, and so those are very important innovations. Using these techniques to improve the varieties is a very important way of helping farmers cope with their challenges. I am in my last slide, but this is where I would like to, I would say this is a distillation of my so many years already. I started working for government in 18 years with the Department of Science and Technology in the Philippines in R&D management, and then move on to a regional agency for agriculture under the ISIBIO, in communicating plant biotechnology. And this slides seem for me like a distillation of, also I share the same thought of what are the elements in the environment that could enable plant science innovation. Not only enable, but you would have timely access. I think timely and access is very important to that. So you would see here a lot of bullets, and I just would like to say that these are not exclusive. They impact on one another: political will, science and evidence-based regulation, societal license. So for example, when I worked in the Department of Science and Technology in the Philippines before, then when we tried to craft the crop biotechnology research focusing on our Philippine export winners, and then rice and corn, we have to reach out to our policymakers, not only to our heads of agencies, but even the legislative. We have to make sure that regulations are there, and also risk-based and science-based. And just one thought about regulations, from a biosafety point of view, we have regulations. So for example, Indonesia, they have the regulation as early as 2005. And so over those years, Indonesia has gained a lot of regulatory experience. And with that regulatory experience, you can get to a more comfortable and confident level how to make it more efficient. Societal license is very important. If consumers, so like the GMOs, there's until now, you can still hear a lot of people against it. You can still hear a lot of misinformation. So, and that is making our politicians nervous as well. And that is also making our regulators, slow down or just kind of take a little bit time, because, when there's a lot of noise, that is also affecting the decision making. But here we really have to also listen to our farmers, because farmers need choices. And you are researchers, and not only researchers, when you do into commercializing the products of research, we have to increase our investment. Public investment to research must be sustained and must be increased. IP protection, intellectual property. This is very important because when you invest in research, you also want to gain something from your investment. And the last one here is public-private partnership. Whether it's a global event or global agreements, public-private partnership is important because nobody can exist as an island. And so we're hoping that, CropLife Indonesia has been, all these elements, they've been working on this, engaging our governments, engaging the different stakeholders, that someday we can add more choices to the crops that are available for farmers, not just GM sugarcane, not just GM potato, but also GM corn and the other, I heard from Pat Master, the GM tomato and all the other products in research still on gene editing. We really hope that those can come on the ground commercially, that it can be commercialized, be at the hands of our farmers, and inevitably that will redound to the benefits of consumers. So that will be my last parting thought. And with all your help, we can all live in a better planet. Thank you so much for giving us this opportunity.
Thank you, Mrs. Sonita Baba, for the great presentation. Ladies and gentlemen, now we will have a break for an hour and we will start the parallel session at 1:30 p.m. Jakarta time. Thank you.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Halo sahabat LIPI, selamat datang di video tour Biosafety Level 2 dan Biosafety Level 3 LIPI. Lembaga Ilmu Pengetahuan Indonesia atau LIPI merupakan lembaga penelitian yang memiliki fasilitas laboratorium Biosafety Level 2 atau BSL2 dan Biosafety Level 3 atau BSL3 yang terletak di kawasan Cibinong Science Center, Bogor. Laboratorium ini digunakan untuk melaksanakan kegiatan penelitian yang melibatkan organisme dan patogen yang berbahaya. Saat ini, kedua laboratorium tersebut telah ditetapkan sebagai laboratorium pemeriksa COVID-19. Biosafety Level, atau biasa disingkat BSL, adalah suatu tingkat sistem keamanan yang dibuat untuk menangani bahan biologi berbahaya di laboratorium. Tujuannya untuk melindungi personil yang bekerja di dalam laboratorium juga lingkungan dan komunitas sekitar dari paparan mikroba patogen yang sedang diteliti atau diuji. BSL memiliki 4 tingkatan, dari 1 sampai 4, di mana semakin tinggi tingkatannya, maka semakin khusus fasilitasnya, dan semakin berbahaya bahan biologi yang dapat ditangani di fasilitas tersebut. Perbedaan utama dari tingkatan ini adalah sistem tata udara, tekanan, kelengkapan fasilitas, dan alat penunjang lainnya. Kita ke laboratorium BSL-2 dulu yuk! Di dalam BSL-2 ini, ada banyak ruangan yang digunakan sesuai fungsinya untuk deteksi virus SARS-CoV-2 sebagai penyebab pandemi COVID-19. Ruangannya terdiri dari 1. Ruang penerimaan spesimen. 2. Ruang logistik sebagai tempat penyimpanan bahan kimia dan bahan habis pakai. 3. Ruang ganti untuk pemakaian alat pelindung diri. 4. Lab ekstraksi yang terdiri atas ruang antara, ruang laboratorium, ruang mandi, dan ruang dekontaminasi limbah. 5. Lab PCR yang terdiri atas ruang antara, ruang pre-PCR, ruang post-PCR, dan ruang analisis. Nah, kalau tadi sahabat LIPI sudah ke BSL-2, yuk sekarang kita ke BSL-3. Fasilitas laboratorium BSL-3 LIPI memiliki infrastruktur yang lengkap dan telah tersertifikasi sesuai standar WHO sejak tahun 2018 dan terus diperbaharui setiap tahun. Biosafety Level 3 memiliki 4 laboratorium, yaitu Clean Rooms, yang terdiri dari Health Biotechnology Laboratory, Food Biotechnology Laboratory, Biosafety Lab, terdiri dari BSL-3 Laboratory, Animal BSL-3 Laboratory. BSL-3 LIPI terdiri atas 2 ruangan. 1. Ruangan mikroba. Ruangan ini digunakan memperbanyak atau mengisolasi materi genetik bakteri untuk digunakan dalam kegiatan penelitian. Misalnya, pencarian obat baru, vaksin, dan sebagainya. 2. Ruangan kultur sel. Ruangan ini digunakan untuk mengkultivasi virus di dalam sel mamalia hidup. Sedangkan Animal BSL-3 terdiri atas 2 ruangan. 1. Ruangan isocages. Ruangan ini digunakan untuk memelihara hewan pengerat yang digunakan sebagai model pada uji di laboratorium. 2. Ruangan pembedahan. Ruangan ini digunakan untuk melakukan pembedahan hewan dan mengambil sampel organ tubuh dari hewan. Nah, untuk mendukung pemerintah dalam mempercepat penanganan COVID-19 di masa pandemi, ruangan di Animal BSL-3 ini dialihfungsikan untuk mendeteksi COVID-19. Sehingga, semua ruangan di Lab BSL-3 digunakan secara aktif untuk semua kegiatan deteksi dan riset SARS-CoV-2. Seluruh ruangan di BSL-2 dan BSL-3 dilengkapi dengan berbagai peralatan yang mendukung aktivitas penelitian, juga didukung oleh sumber daya manusia yang terdiri atas para peneliti dan prekayasa terlatih, tim logistik, dan tim administrasi. Melalui fasilitas laboratorium BSL-2 dan BSL-3 yang dimiliki, LIPI siap berkontribusi secara maksimal untuk mendukung pemerintah dalam mempercepat penanganan COVID-19. Sahabat LIPI dimanapun berada, jangan lengah, jangan menyerah, dan jangan lupa untuk selalu mematuhi protokol kesehatan dimanapun berada ya. Bersama, kita bisa melawan pandemi ini. Salam sehat dan semangat ya!
Ladies and gentlemen, welcome back to ISIBIO 2021. In the following hour, we will have the first parallel session that are divided into 3 breakout rooms. Room 1 and Room 2 will begin with the invited speakers. Room 1 with Professor Anuraga Jayanegara with the topic Modification of Polyunsaturated Fatty Acid Metabolism in the rumen and their deposition in animal products by dietary polyphenol. At Room 2, we'll have Dr. Yopi with the topic Center of Excellence for Integrated Biorefinery as a platform for developing Indonesia's biotechnology research in the future. And at Room 3, the session with the invited speaker will start at 2:30 p.m. Jakarta time with Professor Nicole J. De Vogt with the topic Sponge as Essential Elements in Tropical Reef Ecosystem. Parallel Session 2 will start at 2:50 p.m. and the parallel Session 3 at 5 p.m. Jakarta time. And all the presentation of parallel session are scheduled according to the latest rundown shared by the committee. And to enter the room, choose the square icon, breakout room, on the toolbar below and click join next to the room that you choose. Now you might enter the breakout rooms. Thank you.
Mbak Wiwit, saya di nomor berapa ya? Mbak Wiwit. Halo.
Pak Yopi, Bapak di room 2, Pak.
Oh, saya masuk ke room 2 ya? Sorry.
Betul, Pak. Ya.
Good afternoon, Bapak Ibu, participants, moderators. Welcome to ISIBIO 2021. I'm Naro. I will be here accompanied by my colleagues, Mbak Penny, to accompany the moderators and the participants during the parallel sessions at room 3. So before we start, let me share a screen to introduce the moderator for today's sessions. So I will be reading and introducing the 3 moderators for today's sessions at room 3. Started from Dr. Anggia, followed by Dr. Sugiono, and then the last is Dr. Linda Sukmarini. So let me share it first. As you may see here in the screen, the session 1 at room 3 will be moderated by Dr. Anggia Prasetyo Putri, PhD. She graduated from Institute for Molecular Bioscience, the University of Queensland, Australia, in 2020. She is now the researcher at Center for Drug Discovery and Development at Research Center for Biotechnology, BRIN. She is interested and has been doing research in the area of microbiology, microbial genomics, antimicrobial resistance, molecular biology, antimicrobial drug discovery. In the second sessions will be moderated by Dr. Sugiono, PhD. Dr. Sugiono received his degree, a doctoral degree at Department of Molecular Biology and Biotechnology, University of Sheffield, England, in 2001. He is now a faculty member at Department of Biology, Jenderal Sudirman University, Central Java, Indonesia. He is interested in plant biotechnology and plant tissue culture. And the last moderator will be Dr. Ernat Linda Sukmarini. Dr. Ernat Linda Sukmarini received his doctoral degree at the Berlin International Graduate School of Natural Science and Engineering, Faculty of Chemistry, Technische Universität Berlin in 2019. She is also now a researcher at the Center for Drug Discovery and Development. It's the Center for Biotechnology, BRIN. She is interested in microbial natural products, based drug discovery, and peptide-based natural products engineering. So I think the three moderators have been introduced to you, Bapak Ibu, all participants, and the first sessions will be moderated by Dr. Anggia Prasetyo Putri. Dr. Anggia, time is yours. Thank you.
Okay, thank you very much for the introduction, Dr. Naran. So, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen, and welcome to our very first parallel session of the day. So, at this session, we will have 5 speakers. So, we have the first one would be Ms. Akhirta Atikana from the Research Center for Biotechnology, BRIN. And next we have Bu Santi Puspivasari from the Indonesian Rubber Research Institute, and followed by Dr. Farozi, Mas Eri Septiana, and also Pak Bustanu Salam, all from the Research Center for Biotechnology, BRIN. So first of all, I'd like to confirm that every one of our speakers are here. It looks like they are, except for but I'm sure because he's the last session, that shouldn't be a problem. So anyway, I would like to remind all of our speakers that you all each have 10 minutes for presentation. So that will be for the video presentation. And after all of the presenters are finished, we will have a question and answer session. So a discussion session, approximately about 10 minutes. So hopefully we'll be on time. So there will be some time for discussion later. And anyone who has any questions are welcome to jot down your questions in the chat feature, and they will be addressed later in the Q&A session. So, okay, and without further ado, I'm guessing the committee is ready. I would like to welcome our first speaker, or presentation rather, from Ms. Akhirta Atikana, and time is yours. Guess we'll just wait for the video to show up. There's a little slight technical glitch here, everybody. Shouldn't be a problem. Hopefully we will be up and running soon. So so this session we have a variety of speakers and topics, mostly on drug discovery from natural products. And we also have one presentation from the Indonesian Rubber Research Institute that would be talking about the characteristics that are being considered for replacing imported materials with what can be obtained from Indonesia. So that's actually a really interesting presentation, I'm hoping. And yes, so I hope you all have lots of questions for our speakers today. And now we can begin our first session. Thank you.
Please wait, Bapak Ibu. Thank you, Dr. Anggia. Please wait.
That's okay. These things happen, so it's okay.
Would it possible for the participant to present their own presentation rather than controlling, centrally? So, to avoid the delay, avoid the unnecessary technical glitch like this.
I'm not sure what the committee would think about that, but I assume since everyone's already know that they will have a video on, so they might not be ready for the live presentation. But I guess let's just hope that this will be coming up soon.
Good day, everyone. First of all, thank
Okay,
you
thank
for the
you.
committee of ISIBIO for the opportunity for us to present our experiment entitled Selection of Growth Media to Produce Antimicrobial Compound from Indonesian Actinomycetes. My name is Akhirta Atikana. Here I represent our team. As for the introduction, you know that nowadays the number of multi-drug resistance microorganisms has increasing and become a world problem because it become a threat for therapeutics, cause high mortality rates, decrease the effectiveness of drugs that become an easy target for immunocompromised condition. It prolong the illness, so it also have a high medical cost. However, the discovery of new antibiotics were scarce and remain limited, as you can see here also in the diagram that shows antibacterial drug discovery and the resistance development from 1900 until nowadays 2020, in which the first antimicrobial drug was discovered that also in line with the resistance of many bacteria. However, no new class of antibiotic was discovered recently. We know that actinomycetes are a potential producer for bioactive compounds, including the Indonesian actinomycetes that are well known as producers of many important compounds such as cellulose as well as the antimicrobials. So here in this experiment, we investigate marine actinomycetes from Indonesia. We do screening for their antimicrobial activity, and we select the potential media for further bioactive compound productions. In this experiment, we use 18 marine actinomycetes we revive from glycerol stocks, use SYP agar medium, and then we do selection of different growth media for antimicrobial activity. Using agar plug method, we use the R5, EM, SFM, and OM media that different in their carbon source. So we also do molecular identification for the actinomycetes that showed antimicrobial activity. We found in this experiment our actinomycetes in 4 different growth media showed bioactivity against 3 indicator strain, Escherichia coli, Staphylococcus aureus, and Bacillus subtilis, with the highest number of actinomycetes with antimicrobial activity in medium EM, as you can see here in the graph, with the lowest number in medium R5. So we thought that this is because the media have more complex sugar or malt. In this EM media, it triggers the production of bioactive compounds because it needs more to degrade the complex sugars. So for the R5, it contains more simple sugars such as glucose and sucrose. So in here, maybe there are a carbon catabolite repression happening here, so they did not produce any bioactive compounds. In this experiment, we found that most of the actinomycetes produce narrow spectrum of antimicrobial, in which they only inhibit the growth of Staphylococcus aureus and Bacillus subtilis. No matter the media, we use these two BLH324 and BLH142 showed a relatively consistent bioactivity, in which they also showed bioactivity in each of the growth media. The highest bioactivity was in BLH324 against Staphylococcus aureus in media EM. These two BLH324 and BLH142 was in the cluster of Streptomyces. Streptomyces known as a very potential genus for producing bioactive compounds. It harbors many biosynthetic gene clusters encoding many known and unknown bioactive compounds. Here, both of these isolate was in the genus Streptomyces. BLH324 was 100% similar to Streptomyces hydrogenans, while BLH14-2 was 99% similar to Streptomyces griseorubens. And in the genome of these two species, we found that they harbor many biosynthetic gene clusters that are important for further experiments and analysis. So in this experiment, we highlight the potential of Indonesian Streptomyces as producer of antimicrobial compounds, as well as the importance to select appropriate media for bioactive compound production. With that, I would like to end my presentation with gratitude to everyone involved in this experiment, especially for the funding from Lembaga Pengelola Dana Pendidikan and Badan Riset Inovasi Nasional. Also, thank you for everyone who listening to this presentation. Thank you.
Okay, thank you very much, Ms. Akhirta Atikana. Now we shall move on to the next presenter. I'd like to welcome Bu Santi Puspitasari from the Indonesian Biological Research Institute. And we should have the video up and running soon. So, Bu Santi, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for this good opportunity. First of all, I would like to introduce myself. My name is Santi Puspitasari, Biotechnologist of Indonesian Biological Research Institute. Today, I would like to present my paper entitled Determination of Hydrogenated Natural Rubber Bearing Fat Vulcanicide Performance Based on Thermal Oxidation Resistance Characteristics. Elastomeric weight-bearing patch is an outer rubber-based product which is commonly used as isolation system to protect, bridge, and flyover structures from external loads and energy like traffic load and earthquake. Commercial elastomeric weight-bearing plate composed of multi-layer of rubber and steel plate. The rubber used commonly are natural rubber and synthetic rubber like chloroprene. The presence of high content of unsaturated C-double bonds or natural rubber macromolecular chain cause natural rubber product is easily deteriorates under sunlight, ozone, and oxygen exposure. Nowadays, the low thermal oxidative resistance of natural rubber can be covered by petro-based synthetic rubber like chloroprene rubber. The increasing environment and awareness has proposed scientists to modify natural rubber properties. The modification was aimed to improve the thermal oxidative resistance of natural rubber by means of hydrogenation. Hydrogenated natural rubber is specialty natural rubber which is expected as better thermal oxidative resistance compared to natural rubber, but at least equal to chloroprene rubber. Hydrogenated natural rubber is regarded as promising biomaterial to substitute chloroprene rubber in the manufactured rubber goods for outdoor application. The research was aimed to investigate the possibility of hydrogenated natural rubber as alternative-based elastomer in the production of rubber vulcanicide for steel laminate elastomeric bridge-bearing pad at various capacity of production. The methodology applied in the research was rubber compounding. The stages were the mastication of hydrogenated natural rubber and then followed with addition of rubber chemicals which consist of activator, antioxidant, antiozonant, filler, processing aid, accelerator, and vulcanizer. The process continued with remilling and blending. The compound obtained from the compounding stages then vulcanized at hydraulic press machine to produce rubber vulcanicide. The variable applied in the research were compounding capacity at 300, 900, and 2,700 grams of rubber. Then the other variable were test condition based on natural rubber and chloroprene based. And then the test parameter consists of curing characteristic, hardness, tensile properties, compression set, ocean resistance, accelerate UV weathering, and color change. Especially for accelerate UV weathering and color change are not included in Indonesian natural standard. For accelerate aging condition, at natural rubber base was conducted at 70 Celsius for 168 hours. For chloroprene base, it was conducted at 100 Celsius for 70 hours. For ocean resistance test, based on natural rubber test condition, was conducted at 25 pphm ozone concentration for 48 hours, and for chloroprene-based at 150 HM ozone concentration for 100 hours. The result of the curing characteristics was presented at the table. Curing characteristics is the function of rubber compounding capacity. Compounding at capacity of 900 gram rubber short better curing behavior, which indicate by higher standard torques and shortest optimum curing time. Furthermore, compounding at capacity of 300 gram and 2,700 gram shows similar behavior. Then the histogram shows the hardness test result. Hardness value of the hydrogenated natural rubber vulcanisate are slightly increased after aging condition. However, it remains below the Indonesian national standard requirement. The initial hardness was arranged at 60 ± 5 short A, with maximum increasing point at after aging is 10 point for natural rubber and 50 point for chloroprene specification respectively. And this is the result for tensile properties which include tensile strength and elongation at bricks parameter. Indonesian national standard requires minimal tensile strength as 15.5 MPa and change value of tensile strength as maximum 25% for natural rubber and 15% for pure chloroprene aging test condition. Furthermore, Indonesian national standard also requires minimal elongation at brick as 400% natural rubber and 350% for chloroprene rubber. Change value of elongation at brick as maximum 25% for natural rubber and 40% for chloroprene aging test condition. From the both histogram, we can see that only hydrogenated natural rubber vulcanisate was produced from compounding capacity at 300 gram per batch, appropriate to the Indonesian national standard. Low tensile properties value were expected due to the cross-link network of hydrogenated natural rubber macromolecular were formed by short molecular chain. And this is the result for comparison test based on chloroprene aging condition resulted relatively higher value compared to natural rubber aging condition. It was due to the higher temperature aging condition at chloroprene base, which caused the hydrogenated natural rubber vulcanisate lost the ability to resist deformation generated by compression load. And the picture shows the ozone resistance test result. For the hydrogen natural rubber base test, there was no crack on the hydrogenated natural rubber vulcanisate surface, while for the chloroprene base, there was a crack on hydrogenated natural rubber vulcanisate surface. But similar to ozone resistance test, the result of UV weathering test also showed the absence of damage on hydrogenated natural rubber vulcanisate surface and less color change. We can conclude from the presentation that hydrogenated natural rubber was recommended to be used as alternative-based elastomer for rubber composite tire on elastomeric bridge-bearing pad manufacture. The hydrogenated natural rubber compound formula, which was applied in the research, could produce hydrogenated natural rubber vulcanisate with high hardness level and good thermal properties at any compounding capacity. The hardness and ozone resistance value fulfill quality standard requirement of natural rubber bearing pads in accordance to Indonesian national standard 3967 2013 for Euro 60. This evidence was unfollowed with good tensile properties and compression set value. There is designing hydrogen natural rubber compound formula that was expected to increase the weakness of hydrogenated natural rubber bearing pad vulcanisate in order to substitute chloroprene rubber as base elastomer. Thank you for your kind attention, and we're looking forward for your suggestion. Thank you.
Thank you very much, Ms. Santi Puspitasari. Now we shall move on to our next presentation, which is from Ms. Eri Septiana from the Research Center for Biotechnology, BRIM. We should have this video up and running soon. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Thank you, moderator, for the opportunity that has been given to me. Good morning, everyone. My name is Eri Septiana. I'm from the Research Center for Biotechnology, National Research and Innovation Agency, BRIN. Today, I would like to present our research entitled The Impact of Extraction Method and Solvent on Biological Activities of Garlic Extract. Garlic is a tuber plant that has been widely grown and used throughout the world. This plant is traditionally used as a spice in cooking, salads, and as medicine. In its role in medicine, garlic is reported to have activity as antioxidant, anti-cancer, antimicrobial, and also antihypertensive. This ability is related to the content of chemical compounds like organosulfur and phenolic compounds such as flavonoid. The choice of extraction method and solvent use will affect the biological activity of the extract. Therefore, in this study, we tried to determine the bioactivity of garlic extract using different extraction method and solvent. The activities we carried out in this study include extraction, antioxidant assay. We use TX, PrEP, NO, and TPPH scavenging method, measurement of total phenolic and flavonoid content. In vitro antibacterial, we use Bacillus subtilis, Staphylococcus aureus, and Escherichia coli as a bacterial test. Anti-cancer in vitro assay, we use breast cancer cell MDA MB231. And antihypertensive assay, we use in vitro angiotensin converting enzyme inhibition assay. And the last of which was data analysis. In the yield result, obtained data that the extraction method and the type of solvent have an effect. The ultrasound-assisted extraction method and 70% ethanol solvent have higher yields than the maceration and water solvent method. The compounds in garlic are more attracted to 70% ethanol solvent than water. The combination of water and organic solvent can facilitate the binding of compounds that are soluble in the ethanol and or water. Physical and chemical properties of material treated with ultrasound will change because the propagation and interaction of sound waves will damage plant cell wall. In antioxidant assay, the extraction method and the type of solvent did not affect the PrEP method. In the TAG method, 70% ethanol solvent gave better result than water. In NO scavenging method, in the same solvent, the maceration method gave better result than the ultrasound-assisted method. Meanwhile, in the TPPH method, the effect is only seen in the water solvent, where the maceration method has better antioxidant activity than 70% ethanol. The type of solvent and extraction technique gave varying results depending on the method used on antioxidant activity. However, in general, the use of ethanol as a solvent has better antioxidant activity than water. In the measurement of total phenolic and flavonoid content, extraction technique and type of solvent had no effect on total phenolic content of garlic extract. The type of solvent has an effect on the total flavonoid content compared to the extraction technique. We can see that water solvent is better than 70% ethanol. In antibacterial activity, garlic extract was only able to inhibit the growth of Bacillus subtilis bacteria. Extraction technique and type of solvent had no statistically significant effect on the antibacterial activity of garlic extract. The compound thought to play a role in the activity of garlic extract as an antibacterial is allicin. Allicin will react with thiol groups and can deactivate essential enzymes in bacteria. The anti-cancer and antihypertensive activities were only carried out on extract with ethanol solvent. Extraction technique using ultrasound-assisted has better anti-cancer and antihypertensive activity than maceration in the same type of solvent. The ability as an antioxidant is closely related to anti-cancer and antihypertensive activity. Because the reactive oxygen species, or ROS, production can affect the regulation of vascular tone by increasing vasoconstriction of blood vessels. The increase in ROS is usually accompanied by the activation of oncogenes, which is the first step in the malignant transformation of cancer cell. Therefore, extract with the ability as antioxidant and anti-cancer as well as antihypertensive are very good to be developed. From the result obtained, it can be concluded that the type of solvent and the extraction technique gave varying results depending on the method used on antioxidant activity. However, in general, the use of ethanol as a solvent has better antioxidant activity than water. Extraction technique and type of solvent had no statistically significant effect on the antibacterial activity and total phenolic content of garlic extract. Ultrasound-assisted extraction method and ethanol as a solvent has produced more yield and generate better anti-cancer and antihypertensive activity than maceration. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Okay. Thank you very much, Ms. Eri Septiana. And now we shall pay our attention to the presentation of our next speaker, Dr. Fahru Razi. And we should have his video on soon. Thank you.
Thank you, moderator, and good afternoon, ladies and gentlemen. I am Fahru Razi from Research Center for Biotechnology, and today I would like to present our research concerning the exploration of antifungal activity from seed coat slime associated with bacteria. First, as an introduction, let me introduce you a little bit concerning the seed coat slime. In the red side here is the seed coat slime, and in the cocoa bean fermentation is commonly known that after 2 days of the fermentation, the alcoholic fermentation by yeast was decreasing. But up to now, we don't know yet why the alcoholic fermentation during the fermentation was decreasing. The previous study was argued that the decreasing of yeast during the second day of the fermentation was caused by the interaction between the heat, ethanol, and also acid was produced during the fermentation. But recently we found that the decreasing of the yeast during the second day of the fermentation was affected by the antifungal protein containing in seed coat slime. And this is our previous study concerning the sequencing, amino acid sequencing of seed coat slime. At least we found that 4 kinds of antifungal protein, namely is osmotin with 24 kilodalton, basic chitinase with 28 kilodalton, carrot chitinase with 31 kilodalton, and glucan endo 1.3 beta glucosidase with 33 kilodalton. And all of this protein, we assume that they can inhibit growth of yeast during the second day of fermentation. An interesting question for me and maybe for us is from where the antifungal protein was come from. So first we assume that this antifungal protein was coming from the sitcut slime itself, and also maybe the metabolite of a microbe that active during the cocoa bean fermentation. For example, maybe from fungal and also maybe from bacteria. And today I will more focus my talk into the bacteria associated with slime and have also the antifungal activity. And this is the material we use for our research. So first we take the cocoa pod from different place in Indonesia. First we take from the Pandeglang, from Bogor, from Blitar, and also from Lombok. And then normally we take two type of sample. First we use the healthy pot and unhealthy pot, and from Pandeglang we only have a healthy pot, also from Bogor a healthy pot, and from Blitar we have the healthy pot and also unhealthy pot. But from all of this sample, the only sample, cocoa pot sample from Lombok will the healthy status was have the antifungal protein. And this is method we use for the extraction of cocoa sitcut slime. First, from a healthy and unhealthy pot, we open with a knife, and then we will find that this cocoa bean containing also with a pulp. To get the cocoa slime, first we should remove this pulp with the pectinase enzyme for 24 hours, and then we can harvest sitcut slime. And then later this sitcut slime we will use for the isolation of bacteria. And this is method we use for the isolation of the bacteria from sitcut slime. First, from this sitcut slime we make dilution with buffer dilution, and then we format into the agar medium, and then we take a different colony from this first agar, and then we pick up this colony and we do the purification of this isolate. And then after we get the pure isolate, we can use as antifungal assay. And from the first assay, we only use Saccharomyces cerevisiae. And if you have look here, this is the inhibition zone of the bacterial activity, and only IDL2 helps the antifungal activity against Saccharomyces cerevisiae. And to know more about the antifungal activity of IDL2, we also do the assay against 11 different of yeast species and also 3 different species of mold. And then the very interesting result I think for me is the inhibition of yeast with IDL2. Nearly 80% of yeast was inhibited by IDL2. And then interesting research is concerning the inhibition of Candida albicans, Candida parapsilosis, and also Mesenchia pulchirema was more higher inhibited even compared to the positive control we use. I think the antifungal activity from IDL2 and also maybe the antifungal protein from IDL2 was more higher even compared to the control. But in contrast into the mold inhibition, IDL2 only inhibit the Trichoderma species. And then to know what is the IDL2 species, we also do the molecular identification by using the normal primer for bacteria, and we find that the IDL2 was 99% identity to Bacillus subtilis, and we also do the confirmation by using the phylogenetic tree analysis, and then we also find that IDL2 was one cluster with Bacillus subtilis. So we can say that the bacteria containing in seed coat slime who have the antifungal activity was Bacillus subtilis IDL2. So, and this is for our conclusion and maybe for future direction of our research, that the antifungal activity of seed coat slime was limited to a specific variant of cocoa, and it is highly attributed to the presence of the bacteria associated with seed coat slime. The inhibition of Bacillus subtilis show a strong fungicidal effect and broad spectrum against yeast and especially against Candida albicans. The antifungal activity of Bacillus subtilis IDL2 was more active in living cell suspension, and then I think it's more better if we can use for a green fungicide. And for future direction, I think we still need to do the metabolite characterization by using HPLC and also maybe GCMS, and also the optimization method for the antifungal extraction. And that's all for me. Thank you for your attention. And I give back to moderator.
Okay, thank you very much, Dr. Farurozi. And we are now down to our final presenter of the session, which is Pak Bustanus Salam. And the video will be on shortly. So please pay our attention to this presentation. Thank you.
Warahmatullahi wabarakatuh. Ladies and gentlemen, in this ISIBIO seminar, please allow me to share screen. In this seminar, I would like to present our research with the title Anti-Diabetic Activity of Cinnamon Bark Extract Cinnamon burmani Nes Anti-Nes Bro. With the member of this research is Bustanus Salam, Yatri Hapsari, Oji Rahman, Eriseptiana, Partomon Simanjuntak, Kemi Heliawati, and Iin Nopiani. Our research is from Center Biotechnology Lipi Research Center for Semistebrin and Department of Chemistry MIPA, Pakuan University. As we know, cinnamon have a metabolite secondary such as phenylpropanoid, flavonoid, lignan, terpenoid, stearic acid, and steroid. And this metabolite secondary has activity as anti-diabetic. And Cinnamomum burmani Nes Anti-Nes Bloom, one of Cinnamomum genus cultivated in Indonesia. Bioactivity from Cinnamomum species has reported have an activity as anti-diabetic, antioxidant, antibacterial, anti-malarial, anti-leukemia, and anti-inflammatory. And another bioactivity research can against alpha glucosidase enzyme and alpha amylase enzyme. Based on this research of these studies, cinnamon can act as an anti-diabetic compound. This research purpose is comparing the anti-diabetic activity of 6 regions in Indonesia and identification of anti-diabetic activity compound. The method this research include of extraction, fractionation, anti-diabetic activity test, and identification of active compound. The research is the origin from Aceh, Jambi, Brastagi, Bau-Bau, Gunung Mas, and Ambon from in Indonesia. And Gunung Mas has cinnamon bark has a highest activity anti-diabetic among others. So it was chosen for the next process. Cinnamon Gunung Mas extracted with macerasi with ethanol and separated with partition with hexane and ethyl acetate. And the activity from solvent is ethyl acetate has more active from the other extract. And the ethyl acetate was separated with first column chromatography have 7 fraction, and the fraction is number 4 more active from the other fraction. The fraction 4 is separated with second column chromatography have a 5 fraction, and the fraction 4.3 more active with the others, and the fraction 4.3 compared with acarbose is active as an anti-diabetic activity. And the fraction 4.3 identification by FAR has now have a group of OH alcohol, GS alkane, and GC double bond alkane, G double bond H alkane, and GO alcohol, where this group are present is a sterol structure. And GCMS analysis is have a role anti-diabetic of ethyl acetate extract number is beta cytosterol with a similarity 19.8 and the content of 84.87%. And the active chemical compound fraction 4.3 as known as beta cytosterol is Umar has reported beta cytosterol from Dilenia indica have inhibit enzyme alpha glucosidase and alpha amylase. And Sujata et al. Have reported beta cytosterol can reduce glucose level. And the conclusion from this research is now as ethyl acetate from Gunung Mas cinnamon bark had a highest activity, diabetic activity, and we know as beta cytosterol from FTR and GCMS analysis. In this moment, I would like to say thank you for Research Center for Biotechnology LIPI for research grant from DIPA program in 1917. Thank you very much. Stay healthy, safe, and happy. Have a nice day. Thank you.
Okay, thank you very much, Pak Bustami Salam. So now we have completed all of our 5 presentations, and we have now arrived to the question and answer session. So it is 2:18 at the moment. So we'll have potentially about 10 minutes for discussion for any questions that anyone might have. And since we haven't got any questions in the chat rooms, I would like to invite if anyone would like to ask questions directly to one of our presenters. And could I please ask our presenters to have their video on if possible? Thank you. During the session. And anyone at all? Any questions? Please feel free to raise your hand or unmute and directly ask the question. Okay. Well, I have quite a bit of questions for each of the presenters. So I will ask probably 1 or 2 questions first, if I may. I have a question for the first presenter, Ms. Akhirta Atikana. So you were presenting about the variations in a medium to produce metabolites from actinomycetes. In your knowledge, are there any other approaches that are feasible to be applied for optimizing the production of secondary metabolites in actinomycetes other than the variations in nutrients?
Yes, can you hear me? I think
yes,
my video is not really working.
That's okay. Okay.
Yeah.
All good, we can hear you.
Thank you, Mbak Anggia, for the questions. And as my knowledge, I think that after this selection, because our experiment is like a preliminary selection before we move further. So first we select which media that are the best in our study for producing the bacterial compounds. So for next, I think we can like make a variation on the temperature, for example. If we already select which media that we use, and then we can also check which temperature that are optimal for their production, for examples. And also I think in this temperature and then maybe pH, I think it's also possible. Also, yeah, I think that's what we can do after that.
Okay, so variations in the conditions of the culture, such as pH and temperature. Okay, well, thank you very much for that answer. I'm sure it'll be useful for other people who are also working in similar fields to see how they can better improve the results of their metabolite production of interest. So thank you. And any other questions from the audience? If not, I would like to
actually have a question for Mas Eris.
Okay, well,
yeah.
Please feel free.
In one of your result, you mentioned about the anti-diabetic and anti-cancer in different concentration, if I'm not mistaken. So that means you test both concentration or only that respective concentration in the test that I did not clear from your presentation. Thank you.
Okay, thank you. Can you hear my voice?
Yeah.
Okay, thank you for your question, Mbak Hirda Tikana. In my result, in antihypertensive, not anti-diabetes, in antihypertensive and anti-cancer, we use different concentration for each assay because we did a preliminary screening first to get a good result in anti-cancer and anti-hypertensive. Also, we use different concentration in both assay.
Okay, so that means because I saw like two different concentration in there. So one concentration for anti-cancer and one concentration for anti-diabetic, anti-hypertensive.
Yes.
Okay. All right. So do you think it was like any influence of the concentration to the result, for example? Like if you use different concentration, of course, it will give different result, right?
That's right. Because we use a different concentration, so the result in antihypertensive and anti-cancer is different.
Okay, so what you show here is the concentration that the highest activity, or it's like the IC50 or something?
We did not yet to IC50, just in inhibition concentration.
Okay, all right, thank you.
You're welcome.
Okay, thank you for that. I'd like to welcome anyone else. Should they have any questions to any of our presenters? Anyone?
May I have one?
Yes, please. Feel free. Thank you.
Okay. Thank you very much for the time. And I have question for Ibu Santi Puspitasari. in case for your materials, did you also conduct the TGA measurement? Because I see your title, your research want to study about the thermal oxidative resistance characteristic of your materials. So did you measure that analysis? And the second one, for your material, did you prepare the hydrogenated natural rubber by yourself, or you just buy and then try to mix with the natural rubber? Maybe that one for me.
Please, Busanti. Thank you.
May I answer the question, Ms. Chairman? Okay, thank you for the very interesting question. I try to answer the question. First question is about TGA. Well, in this experiment, I did not conduct the TGA testing because the analysis we limited on refer to the Indonesian national standard, and it's only mechanical and physical test. And the second question is about the hydrogenation. Well, we made the hydrogenated natural rubber at the previous year of the research. So we developed the application at the next year. I think that's the answer. Thank you.
Okay, so maybe just suggestion for you. So maybe I think the TGA material, I mean the TGA analysis, we can get more information about the change of the structure of your material, because if I'm not wrong, when I see your presentations, one of material, if I'm not wrong, they have cracked, right? If I'm not wrong. And then I think if you get the TGA analysis, you can see in which temperature that maybe that material will have some change of the structure. And yeah, of course, this is interesting for you said that you prepare your material, and maybe it is better when maybe it is better if you put the characteristic measurement, like let's say the FTR of your hydrogenated natural rubber, and also compare with unhydrogenated natural rubber. I think that's a suggestion from me. Thank you.
Thank you for the suggestion. Well, I will improve the paper with FTR analysis. Thank you.
Okay, thank you for that, Santi and also Mas Bayu. I think this is why we have these sort of conferences to exchange ideas and have inputs from others who could improve our work. So that's actually very good. So we have about 3 minutes left in the session. And if anyone has one final burning question, please feel free to do so. Just one more. Okay, well, I have one for Mas Farurozi. You mentioned in your final slide that the information that you obtained from this research could potentially be useful for application as a fungicidal for the results that you found. So what sort of application would you be looking at? Would it be isolating the compounds, or would you be using the whole organisms, or what would you be envisioning for that particular application? Thank you. Mas Waji?
Sorry. Yeah, can you? Okay. Yeah. For the application, I think we have two directions. First, I think we will use as antifungal activity during the cocoa bean fermentation, because during the cocoa bean fermentation, I think it's a lot of fungal who have not affected directly into the cocoa bean fermentation, and they make also the how we can say they produce some toxic, and then we want try to inhibit the growth of the fungal who have a toxin into the cocoa bean. And interesting also research in our result is that the Bacillus subtilis of IDL2 was more higher activity in the bacteria itself, not in the protein in the bacteria. So yeah, I think it's better also we can use this bacteria directly to fungicide for preservation of post-harvest of agricultural product, maybe for example apple or something like this. Okay, that's all. Thank you.
Okay, well, thank you for that. Let's hope that we'll get to that stage one day. And the time is now 2:30, so I think it's time for me to conclude the session. So thank you, everyone, all of our speakers, and also the audience for your attention. And I would like to return this session to the organizing committee. So thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Anggia, for hosting session 1. And thank you, Bapak Ibu, for following the sessions. So now we are entering session 2 that will be moderated by Dr. Sugiono from Jenderal Sudirman University, Central Java, Indonesia. As I delivered before in the beginning, that Dr. Sugiono graduated from Department of Molecular Biology and Biotechnology, University of Sheffield, England in 2001. He is really interested in plant biotechnology and plant tissue culture. So by now, he will be moderating the sessions, started from the first speaker in session 2, Professor Nicole, and followed by other speakers in this parallel sessions. So Dr. Sugiono, please, time is yours.
Okay, thank you, Pak Naro. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to take the opportunity to welcome Professor Nicole de Foots for her time. I understand that it is early morning in Amsterdam now. I think it's about 7:30 in the morning. Is that right? Okay.
Yes, that's right.
So it's a great opportunity for us to listen what Professor de Foots have already been doing in the whole of her life. So I just wrote in her short CV that she graduated from University of Amsterdam with her PhD thesis on biodiversity and mariculture potential of Indonesian sponge. So I assume that she knows well about Indonesian marine diversity, for instance. So I also wrote that she is very reputable scientist with H index more than 43, more than 7,000 citation has been get by Professor de Foots. And today she will be speaking on sponge as essential elements on tropical reef ecosystem. So without further delay, I would like to welcome Professor de Foots to present her idea. Professor de Foots, the time is yours, and you have 20 minutes for that. Thank you. The time is yours.
Thank you for this introduction, and I will start sharing my screen. Okay, I hope I have enough time, so I will talk a little bit fast, if you don't mind. Many of you probably will know that I love sponges, and I've been working on sponges for a long time, and I have actually started working in Indonesia as a student and also as a PhD student. And currently, let me see. I work at Naturalis Naturalis Biodiversity Center, and since 2005, And here you see our new museum. This opened last year, and unfortunately we had to close down for about a year because of the lockdown, but we are gently opening again. And I'm positioned in a research group, Marine Biodiversity, where we study the past, the present, and the future of marine ecosystems. But of course, today I'm talking about sponges, and maybe some of you will not know, but you probably heard already today that sponges are well known for the prolific production of bioactive compounds, which has gained the interest of many. I'm slightly affiliated with this kind of research because I do a lot of identifications. And why is that? Well, sponges has not been the focus of coral reef research in the past so much, mostly on corals and fishes and algae, and really to a lesser extent to sponges. And it's even hard to see in this image where there is a sponge, but it is there. And the reason for that is that they're quite difficult to identify. If you look at sponges, they are very diverse in life form and lifestyle and lifespan. And here you can see some examples from Indonesian sponges, where you can see encrusting forms like here, and the well-known great giant barrel sponge, and sponges that look a little bit more like corals, if you would say. But sponges are real animals. They're the most primitive multicellular organisms on Earth, but they're real animals. And on the left-hand side, you can see a giant barrel sponge spawning. So you can see the male propagules exuding into the water. And on the right-hand side, you can see the eggs of this same sponge. And then when they find each other, they form a real larvae, and they produce small juvenile sponges. So what do sponges do? They don't do so much. They sit on the ocean bottom, and they occur actually in all water bodies on Earth, and not only in marine systems but also in freshwater systems. There's not so much known about freshwater sponges in Indonesia, so I think that would be a very interesting subject to study as well. But what they do, they actively attract a water current, and it enters through these very small openings here, and then they enter these small chambers. So here's an enlargement of that chamber. So the water flow go in, and then they take out bacteria, particulate organic matter. Since shortly, we also know that they can take in dissolved organic matter, DOM, and the waste product is exhaled through the large opening called an oxculum. The lifespan of the sponges is quite different. We also don't know so much about the growth rates of different sponge species, but we know that some species, like the shallow water sponge in Indonesia, that it can grow really fast, probably a few centimeters a week, but also other species are slow growing, even as slow as corals do, and they become very old. So this species, the giant barrel sponge, they have found specimens of over 2,000 years old. So that is also quite interesting if you're looking at sponge research. Sponges are also important in the coral reef ecosystem, so they provide food, but also a home. Here you can see a variety of different animals that live in and inside sponges. These are all examples from Indonesia. So this crab was described from Bali not too long ago. These are polychaete worms that live inside the canals of the sponges. It was described about 15 years ago from Sulawesi. And here you can see sea cucumbers, which possibly we believe feed on the poop of sponges, so on the exudates of sponges. Well, since short, we know that sponges are extremely important on reef systems. Of course, we know that they're important for their filtering capacity. They did a research in the 1970s where they removed all the sponges from the reef systems, and within a few days' time, most of the organisms that live in the reef were infected with bacteria and viral infections. But what is new, and that is actually discovered by a colleague of mine from the University of Amsterdam, from Jasper de Goey, is that one of the greatest paradoxes from Darwin is that he didn't understand that you could have such a enormous diversity of these coral reefs, as all those colors living in an ocean that is so nutrient poor. And what they discovered is that sponges, I already told it before, but most of the, I hear some noise in the background. Maybe the person that is producing the sound can mute. Mute. Okay, so that's okay. It was just distracting me. But many of the nutrients that are in the oceans are dissolved organic matter, so we call that DOM. And these nutrients are inaccessible to most of the animals that live in coral reef systems. But what sponges can do, they can take in dissolved organic matter, and then they produce, well, basically sponge poop. So that is the particulate organic matter. And that becomes available again to other animals in the reef and also to the corals. So they're actually more important and actually the drivers of many marine ecosystems because of this. But where I come in is that sponges are extremely difficult to identify in the fields. Many of you maybe send me images from sponges to try to identify, but unfortunately, most of them I cannot identify from pictures. And here you can see some images that I took from Pulau Pramuka in the Thousand Islands. And looking at those, just at the image, now I know what they are, but when I collected them, I also didn't know what they are. So I have to take them into the lab. And here you can see the 19th floor of the Naturalis collection in Leiden. And we have many thousands of sponges. And so what we do, I take it into the lab. I have to take every sponge that I receive into the lab. I have to cut a small sponge from the skeleton, and then I have to use a microscope. Many of you think that I dissolve the skeleton and I look at the spickles, but that is not enough information for me because I have to look at the orientation of the skeleton of the sponge. And then I take it to my microscope, where you see it here. And I use a lot of literature, and then with a little bit of hope I can identify it. So this is a little bit how it looks like, the sponge skeleton, for those who are interested. So I look at these tiny little structures, they're called spickles. And please pay attention to this small one. But most of the time, the magnification of the normal microscope is not enough. And I take it down to the scanning electronic microscopy, where I can enlarge up till 15,000, 20,000 times, and then I can really see the structure. And this gives me the information to identify. So it's really taking a lot of time to identify sponges, and there are not so many people willing to do so. And I have to say, it's also quite difficult. It takes a lot of time and patience to really understand a little bit. And so I know that, now there are some sponge taxonomists in Indonesia. I'm very happy about that. And so please try also to enthusiasm, because I cannot do all the work. It's too much. So here are some of those spickles, how they look like through the microscope. So the different morphology, so you actually see different morphologies, give me an information, and in this case, at the species level. And sometimes they can be really beautiful, as you can see here. And also the difference in the sizes of those spickles is enormous. So, but sponges has not become the subject of research for their bioactive compounds. Sponges have become a major problem in Caribbean reefs, as you can see here. This is an example of a Caribbean coral reef. And for you that study coral reefs will see immediately that there is no coral in the picture. So they're mostly composed of sponges. And also bio-rowing sponges are increasing in abundance in the Caribbean. So this was my interest in the past years, if we could also find so-called sponge reefs in the Pacific. Well, the theory behind this is that they think they're not, because on the left-hand side you can see how a Caribbean reef looks like and how a Indo-Pacific reef looks like. So in general, the coral reefs, there have less diversity in the Caribbean, and also in a natural way, there are more sponges, they're larger, and also more algae, and that is because of the rivers that come into the Caribbean Sea. And in general, there are more corals and less sponges in the Pacific. Here you can see how that looks like. So in the black bars, you can see the algae abundance in cover in 2 Caribbean reefs in the Florida, also the Florida Keys, and an example from the Great Barrier Reef here, where you can see that coral cover is much higher here. So I will have to go a little bit fast. So I've been wondering how to study this because we don't have a lot of baseline studies in Indonesia in general. So I was lucky enough to travel to Ternate in 2009. And what is interesting from that is that a German professor visited Ternate in the 19th century, and he collected a lot of sponges from that region. And this was, well, you all know where Ternate is, of course, in Indonesia, but here in Europe, no one knows. Here you can see the sites that we visited in 2009. So what I've tried to do, I would try to rediscover the species from Chile, because this is a really important work for me. The diversity of sponges was mostly described from Indonesia by Chile. So I also went to visit the Berlin Museum, where you can see these small samples which I had used to link the specimens that I found in Ternate. And some of them I did find. So this black species here is actually hiding under this blue species over here. And I also discovered this species here, which is apparently a pest in Hawaii. They think it's an invasive species, but I proved it's wrong. And so it was a big puzzle, very complicated, but eventually I came off with this data. And here you can see the number of species, because of course I cannot say anything about the abundance. So what I found is that there was a big discrepancy in the number of species when you look at the order level. So for instance, when you look at the order of the Haplosclerida, so it's a large order with many colorful species, I found much more species in 2009 that was observed in 1896. But also for this group that doesn't exist anymore, but there were more in the 19th century than I found. And some of the groups were not recorded, for instance, in the 19th century. So how did that work? Well, one of my conclusions was it must be due to anthropogenic stress. There was a lot of altered habitats, also deforestation, siltation, and pollution in the area. But also many of the observed littoral sponges were not found in 2009. This was probably due to habitat disturbance or natural species turnover. I don't know exactly why. But of course, there was a difference in sampling method and effort. Because I also sampled the deeper reef sites, and also I sampled not just Ternate, but also around Ternate. And also I didn't find the species and communication curve yet. Yeah, I'm going a little bit fast, but I want to show you all the stuff that I've done. So this is an example of a historical example, which we don't have so much from Indonesia. And I said before, I started working in Indonesia in 1997 in the Spermont archipelago near Sulawesi. And we returned there every 5 to 10 years. And we were actually supposed to visit the Spermont last year. Of course, we couldn't. We were hoping to come this year. Again, we couldn't. And we're really keeping our fingers crossed that we come and visit next year. But in the green circles, you can see which sites we have visited, multiple sites. And this is how the reefs look like. Some of them, this is one of the locations with a high coral cover. However, the diversity is not so high and there's no fish. Also, there has been some bleaching events due to warming ocean temperatures, but also the effects of dynamite fishing. So many of the reefs, they don't look so good. And also some recent crown of thorns attacks killed many of the corals. That sounds all very depressing, but still we found a lot of corals. And what I found is when you compare all the sites that I visited between 1997 and 2018, is that the common sponges were still common. So you could still see those really about the species that actually these occur throughout the Indonesian archipelago. So they're really common sponges, but they were still common in the region. And when you're looking at the benthic cover in the region, it still really looked like a proper Indo-Pacific reef. So the coral cover was still relatively high. And when you look, so the other groups are, for instance, rubble, this RS, DCA is dead coral and algae and some other benthic groups. But you can see that the sponges in general, when you look at the complete mean cover throughout the archipelago, the sponge cover is still relatively low. Sponges do not like dust cover, as you can see here. We also found some sponge diseases. But when you look over the years reading the different sites and the depths, there was indeed an increase in sponge cover throughout the year. So shallow means very shallow, and deep is until 15 meters of depth. So there seems to be an increase in sponges, especially in 2018. And this is mostly due to these encrusting species, cyanobacterial sponges. They're becoming very successful in the Pacific Reef. On the left-hand side, you can see an example of the black disease that can be very problematic, as it can overgrow and kill corals very fastly. But also we discovered two other species that look exactly the same as the black disease on the right-hand side. And one of them is a new species, and we're describing it at the moment. And what we discovered is that these species that are becoming really abundant have a very high abundance of presence of cyanobacterial symbionts in their skeleton. And all of them have exactly the same cyanobacteria. So something is going on there. Here you can see the reported sighting of the black disease. So the Latin name is Terpius hocinota, and it was discovered from Guam in 1973, and it's really expanding now throughout the Indonesian archipelago, but also through the Indian Ocean. And we don't exactly understand the mechanism behind this. We think that is due because of the stress that coral endure. And here you can see those other two species that are very similar to Terpius hocinota in the Spergemonde archipelago. And until 2018, so I'm not sure what is going on in the past 2 years, is that it's especially abundant in the mid-zone area of the Spergemonde. So it's not very abundant in the offshore zones, not in the inshore zones, but in the mid-zone where the coral cover is still high, it has become very abundant. But also we discovered that in the shallows, there are presently many sponges. So this used to be a really nice coral reef. Those green things that you see in the middle, they're actually not algae, but they're all sponges. So they're really taking over shallow coral reefs. So could we say that sponges are becoming more successful because of their symbionts? I think they are. I'm running out of time, I think. Let me quick look. So one of my recent research areas has been looking at microbial ecology of sponges. And sponges are divided in two groups. We call them high microbial abundance sponges and low microbial abundance sponges. And those cyanobacterial sponges actually belong to the low microbial abundance sponges. And they're really two different functional groups. So high microbial abundance sponges are in general long-lived. They're large and massive. They have a lower pumping activity because they're depending on their foods mainly because they're endosymbionts. So they have a high diversity and abundance of microbes, and they're specified by the bacterial groups Chloroflexi, Acidobacteria, and Actinobacteria. When you look at low microbial abundance sponges, they're shorter lived, they're mostly small, but they can grow really fast, as I showed you before. And although they have a lower diversity and abundance of microbes, they still depend on foods from their photosymbionts, as you saw. So they're becoming very important. This is a recent article that just came out a few weeks ago, where you can see the conceptual model of 12 common sympatric sponge species in the Caribbean Sea. And what you're seeing in this graph is that the phototrophic sponges, you can see that on the right-hand side, those are the species that really depend on sunlight. So they really, when the coral's coming deeper, those are the sponges that probably can grow very fast. And then you can see the sponges, for instance, the slow-growing animals that are dependent on bacteria. They get most of their food from dissolved organic matter. And the other ones, the filter feeders, get their food from particulate organic matter. So in this way, you can a little bit predict how and which species might increase in the future, depending on what kind of circumstances you have. So one of the important questions that we may ask ourselves, what is the response of these microbial communities in sponges, and what are their underlying processes? And with this, I want to thank you. Terima kasih. I was talking very fast, I know, but I'm also always very enthusiastic. So thank you for your attendance.
Wow, thank you for a very nice presentation, Professor De Vos. We still have around 6 minutes, so I would like to invite our audience to raise their hand if they have any question to Professor De Vos, because I haven't got any question on the chat, so I invite our participants to ask directly in this session. May anybody like to ask any question? If not, I would like to start actually. Pak Asep Bayu, please do so. The time is yours.
Okay, thank you very much for the nice presentation, Professor De Vos. Actually, previously I worked in the Research Center for Oceanography, and I had many discussions with my colleague as marine biologists. And one of interesting point that they have shared to me is about the effect of the climate change into the some marine organisms. So in case for, let's say, for coral, maybe they said that coral, the climate change has a significant effect into the coral growth. And how about the sponge? Do you have some information about this effect on the growth, or something like the chemical profiles of the sponge related with this situation? Maybe that's from me.
Okay, before answering the question, I would like to add actually, according to Pak Bayu question, do you see any significant trend in terms of sponge biodiversity or sponge abundance in relations to the climate change, especially the increasing seawater temperature?
Yeah, so in general, as you all know, is that corals are very easily disturbed by higher temperatures because they lose their photosymbiont symbiodinium. What we know now is that there are some sponge species that also have this symbiosis with symbiodinium, but they apparently, or what they do, they have a different clade. So I've been studying these sponges also in the Spermont archipelago. I published actually an article on that last year. What we discovered is that those sponges that have a symbiosis with symbiodinium, they only occur in the shallow water. So they're living sympatrically with corals. And in case of higher temperatures, when corals lose their symbiodinium, sponges do not. So they have these mechanisms that their symbiosis is better preserved. So that is one. So in that case, and on the other hand, the other species of sponges, they don't have this symbiosis with symbiodinium, but they have this symbiosis with bacteria. And they don't seem to mind the warming temperatures at all. So what is happening, I think, is that it's not that they grow better in warming temperatures, maybe, but they take advantage of the better health, of the worsened health from the corals. And that is what I think what you see in the mid-zone, in the Spermonde archipelago, is that there's still a lot of coral, but they're affected by many things. So not only higher temperatures, but of course also influx from nutrients. And these cyanobacterial sponges, they actually like the higher temperatures. So they can increase in abundance. And I'm not sure if that's really the only mechanism that is going on, but you really see that sponges are not so much affected by ocean acidification or by higher temperatures. And in case of a higher nutrient runoff, they like that because that's nutrients and they filter the water for the nutrients. So in all cases, they opportune from global warming and global other climate changes. And that is maybe what you see more in the Caribbean. So there already, most of the corals has been replaced by sponges. And the difficult thing is to predict which sponges will be the winners of this reef. That is, I think, still very difficult to say because we don't know so much about the historical abundance and presence of sponges. And that is really the reason for that is really because they have not taken into diversity even also LIPI, I think, they have been excluded just because they are so difficult to identify. And even for me, when I go now, because of all my experience, there are many species that I can identify just by looking at them, depending on the region. So if you take me to Jakarta or the Spermonde, I can almost identify all of them. But if you would put me in Papua or Raja Ampat, I wouldn't have a clue. You know, I would recognize the most common sponges. So, but also the algae. So the algae are also a very important player. And when you're looking in those shallow water environments where the temperature is really high, in those waters that I showed, the temperatures can be 33 or 34 degrees, which is actually a prediction for over 60, 70 years for coral reefs. So I think those are the interesting environments or habitats to study. So what can live in those shallow water environments under those circumstances? And then you can a little bit predict or maybe do some transplantation experiments or whatsoever into deeper areas, because those are the organisms that can endure higher temperatures and nutrient runoff. And again, I'm talking a lot, but it's interesting, I think, to really, take sponges more into account in the biodiversity studies. And of course, it's easy because they're so interesting for their bioactive compounds.
Okay, very interesting information and discussion actually, but unfortunately, we ran out of time. So for Bu Tuti, you may contact Prof. De Voorts for some clarification about the result of current expedition that there are, about 850 species increasing in this area. But anyway, we would like to thank Professor De Voorts for your time and your kindness in sharing the important experience and important knowledge that you have. And we also wish you next year you could visit, Indonesia. And I'm sure that the situation is getting better and better. So it might be easier for the next year. Okay, thank you very much for
bye-bye. Thank you.
Okay, bye-bye. Assalamualaikum warahmatullahi wabarakatuh. And I return the session to the organizing committee before proceeding into the next parallel session. Okay, thank you very much. Back to Mas Naro.
Thank you, Dr. Sugiono. Thank you, Professor Nicole. Now we will have another sessions to listen presentations from our partners from PT Wadya Prima Mulya and ITS. So please, Mbak Penny. Please wait, Bapak Ibu. Our team is trying to share the screen. And Professor Nicole, if you don't mind, and if you are still staying here, would you like to answer the questions from Ibu Tuti in the chat box? Thank you, Professor Nicole, if you don't mind.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya.
Wah, cukup banyak ya. Tapi tenang saja, pengujian porcine ini tidak sulit kok.
Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porcine pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah, tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik, tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porcine pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porcine pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3, yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat sampel mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porcine dari kami. Sekian dan terima kasih.
Halo, kali ini saya mau memperkenalkan alat laboratorium dari Syngene. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel elektroforesis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih, sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat, sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethylene bromida atau cybersafe DNA, atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database tipe gel dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Okay, we are done with the videos from our partners. So to continue the next sessions, for the parallel sessions 2, I would like to ask Dr. Sugiono to continue hosting the sessions. Thank you, Dr. Sugiono. Time is yours.
Okay, thank you, Pak Naro. Now we will come to the next parallel session, which will have 3 speakers, Dr. Nanang Yunarto, and then Dr. Yati Hapsari, and Dr. Tuti Murniase. Okay, first of all, I would like to invite Dr. Nanang Yunarto to present his paper on inhibitory activity of Oncaria gambirox extract, ethyl acetate fraction, and catechin isolate on lipase activity. Pak Nanang, are you ready? Okay, Pak Nanang, the time
Assalamualaikum
is yours.
warahmatullahi wabarakatuh. I would like to say thank you for coming to my presentation. It's great to see you all presenter and participant on the 8th International Symposium on Innovative Bioproduction Indonesia on Biotechnology and Bioengineering. Let me introduce myself. My name is Nanang Gunarto and I'm from National Institute of Health Research and Development, Ministry of Health Indonesia. In this opportunity, I would like to present our research about inhibitor activity of Uncaria gambir extract, ethyl acetate fraction, and catechin isolate on lipase activity. We know cardiovascular disease is globally the main cause of death in the world. 17.9 million people were estimated died due to CVD in 2019, or about 32% of total deaths. Almost 45 deaths in cardiovascular disease is caused by coronary heart disease. Risk factor of CVDs, hyperlipidemia, hypertension, DM, diet, high fat and cholesterol, obesity, and lifestyle. High level of total cholesterol, LDL, increased atherosclerosis. Hypercholesterol is influenced by food consumption that contain high cholesterol as well as synthetic cholesterol in that. A compound that capable to inhibit the activity of lipase enzyme is either in synthetic or natural compound. Synthetic compound that often use of cholesterol reducing medicine is statin. A study that conducted by Ren Kumar reported that a long use of statin had first effects such as myalgia, myopathy, hepatotoxic, peripheral neuropathy, diarrhea, and the adverse encourage natural compound invention as an alternative medicinal treatment. Polyphenol is a compound reported as lipase inhibitor. Catechin and its derivatives are polyphenol that active as antihypercholesterol. A plant that contains high catechin is gambir or Uncaria gambir. Studying the scientific evidence as antihypercholesterolemia, a research is carried out with the aim of determining the activity mechanism in lowering cholesterol through in vitro inhibition test of lipase on the extract, ethyl acetate fraction, and isolate catechin of gambir leaf. Our methods, the first step procedure is extract preparation. Gambir leaf is obtained from 50 Kota district, West Sumatra. The fresh gambir leaf are short, washed, and then put in the tank by steam with hot water vapor. The leaf removed and compressed to obtain the filter. The gambir filtered is dried and stored in a stainless steel container, then dried using an oven and 50 degrees Celsius for 20 hours. The next step, catechin isolate, the extract is put in chromatography column and then soaked with ethyl acetate and hexane while stirring for 6 hours. The catechin isolate is concentrated in ethyl acetate air rotary evaporator at 40 degrees Celsius. Catechin analysis using HPLC, quantitative analysis of extract, fraction, and isolate sample is analyzed utilizing HPLC by UV detection at 280 nanometer using PDA detector. The mobile pass use mixture of acetonitrile and water. Lipase inhibitor activity aside, the plan is determined based on ELISA measurement. The concentration of lipase, the reaction is incubated at 37 degrees Celsius and absorbance was measured at 570 nanometer after 10 minutes. Orlistat is used as positive control and we distill water as negative control. Catechin level in extract and fraction isolate Gambir leaf was calculated by first method catechin standard calibration curve equation. The absorption spectrum showed that catechin were at 280 nanometer wavelength with 14.601 minutes retention time. Measurement of catechin level in extract and isolate catechin of Gambir leaf using the calibration curve equation above the catechin level in the ethyl acetate fraction, isolate fraction, and extract fraction is then 68.32%, 92.18%, and 96.74% respectively. And the lipase inhibitor activity, the in vitro assessment inhibition of lipase activity indicate catechin isolate of Oncaria gambir was the most potent inhibitor of lipase. After ethyl acetate fraction, supernatant, and while extract and olistate show relative less inhibition. Catechin isolate from Oncaria gambir show strong inhibition of lipase activity. The conclusion in this research, gambir extract, ethyl acetate fraction, and catechin isolate were active to inhibit the lipase enzyme in lipid synthesis. Catechin isolate from Oncaria gambir show strong inhibition of lipase activity. The study saw that the higher the level of catechin compound, the greater the inhibition potential of the lipase enzyme. Thank you very much for your great attention. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Pak Nanang, for the nice presentation. And now I would like to invite the second presenter, which will be Dr. Yati Hapsari, who will be presenting a paper entitled with Caulerpa lentifera as a potential nutraceutical resources. Dr. Hapsari, 10 minutes for your time. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you so much for the opportunity so I can deliver our research in this conference. The title of the research is Caulerpa lentifera as a potential nutraceutical resource. And this is the member of our team research. Indonesia has great marine biodiversity such as mangroves, coral reef, and seaweed. Seaweed has been utilized for thousands of years as food and other benefits, for example, for medication, especially in Japan and China. Indonesia has numerous marine algae were consumed as food and medicinal plants. Caulerpa lentifera was one of seaweed from Takalar that has both benefits as food and medicine. It has been studied that nutritional value of seaweed can differ broadly, depends on environmental condition, taxon, and harvesting season leads to various biochemical, tufa, vitamins, and bioactive antioxidant. As plant-derived food, the seagrass will be advantage to solve some health issues. As nutraceutical resource, Caulerpa lentifera can be potential net supplement to increase its benefit content instead only as food. In this research, water and ethyl extract derived from Caulerpa lentifera originated from Takalar, South Sulawesi was investigated its yield, antioxidant, and cytotoxicity activities. Antioxidant activities was carried out using ABTS assay, mineral cytotoxicity activities was carried out using BSLT assay. In general, method of this research are extraction using maceration method and water, and ethanol as solvent, antioxidant assay using ABTS assay, Cytotoxicity assay using BSLT assay and metabolic profiling using GCMS Shimatsu GC 2010 plus series. Result and discussion from yield of extract from Chlorella lentilifera showed that yield of ethanol extract was higher than water extract, although both solvents have similar polarity but have different ability to extract compound from cell. In this case, even cells. Ethanol has higher penetration to extract compound than water. There are numerous methods of extraction such as percolation, diffusion, decoction, and hot continuous extraction, and also parameters such as solvent type, solvent concentration, duration of extraction, and temperature of extraction, which can be optimized to gain better yield and biological activity. Maceration method was selected in order to isolate volatile and non-volatile compounds, although it spends more time than any other methods. Ethanol and water are selected as solvent due to regulation of Drug and Food Supervisory Body in Indonesia, or BPOM. Cytotoxic assay or BSLT assay was a preliminary screening for anti-cancer, and its low cost and rapid screening for investigating compound was preferable than any other assay. From this table, water extract of Chlorella lentilifera showed no toxicity against larvae of Artemia salina, but ethanol extract had cytotoxicity activity against larvae of Artemia salina. An explorating lead drug compound, a rapid screening for toxicity activity of plant extracts need to be conducted before further step using cancer cell line. BSLT assay has been studied that has positive relationship with numerous tumor cell lines. If LC50 was less than 1000 ppm, it showed that extract has cytotoxicity activities. Further research concerning anti-cancer activities can be done for next research. Antioxidant assay using ABTS assay and ascorbic acid as positive control. ABTS assay was selected than other assays because of its solubility in polar and non-polar solvent and stability in broad range of pH. From this, we can see that the value of IC50 showed that ethanol extract has higher antioxidant activities compared to water extract. Ethanol can extract compound that has higher activities than water. As discovered in habitat of light and oxygen that support to produce radical and oxidation compound, Chlorella lentilifera may have durable antioxidant system to defend from oxidative damage. As an edible sea grapes, Chlorella lentilifera has been studied as nutritional content fatty acid in major nutrient in its chemical composition. Secondary metabolites from both extract were analyzed using NIST 14 library. GCMS was selected to identify fatty acid in water and ethanol extract, which preferable solvent for food. Based on GCMS chromatogram of water extract of Chlorella lentilifera was obtained 13 peaks of compounds which classified as terpenes, phenol, and fatty acids. And this is similarity above 90%. Our chromatogram of ethanol extract showed 39 peaks, which can be classified as terpenes, phenol, fatty acids, and esters. Previous research showed that the terpenes have bioactivities as antibacterial, antidiuretic, anti-inflammatory, and anti-cancer activities. Phenol was not just considered as a safe and economical food supplement, but also had potential bioactivities as antioxidant, anti-allergic, and anti-inflammatory activities. So, unsaturated fatty acid is more reactive as antioxidant agent compared to saturated fatty acids due to its ability to oxidize. Both data that shows previous and now were compounds that have similarity index above 90% in water and ethanol. Conclusion of The conclusion of this research, ethanol extract show higher yield, antioxidant, and cytotoxicity activities than water extract of Cholera lentivifera from Takalar, South Sulawesi, Indonesia. Chemical composition of ethanol extract of Cholera lentivifera can be potential nutraceutical resource due to fatty acid as a major compound. Extraction techniques can be optimized for next research to gain better yield and biological activities. I think that's the presentation of my research. Thank you so much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much, Dr. Hapsari, for the presentation. And now we will move to the third speaker, which will be Dr. Tuti Moniase, entitled with Evaluation of Anti-Cancer Activity Against Breast Cancer Cells of Microalga Extracts Collected from Eastern Indonesia. Dr. Moniase, 10 minutes of time is yours.
Hello everybody. Today I would like to present our works with the topics of the evaluation of anti-cancer activity against breast cancer cells of macroalgae extracts collected from eastern of Indonesia. And this is the outlines. And we will go into the introductions. We know that the cancer cases in Indonesia is a high. From the 2070 million population in 2019, 348,000 people is survived of the cancers. And almost 60% of them is death. This is a big problem to the Indonesians. And the most cases of the cancers, the highest cases is the breast cancers. And this is will increasing year by year. And is to be our homework to find new drugs, the effective drug against the breast cancers. The marine biotas, like the animals and plants, we all know that this is a very potential for the new candidate of the drugs, especially for the cancer drugs. And we will choose in this study with the macroalgae. Why we choose the macroalgae? Because we know that macroalgae, most of the macroalgae is edible, and also known some of the species already have some potential as the food supplement, also the drugs. And about the potential of macroalgae indicator, the previous study reported that there are some brominated compounds like the bis2,3-dibromophosph-dihydroxyphenyl that's very active against HeLa cell lines. Also several cell lines like the human hepatoma cells with the IC50 or less than 20 microgram per milliliter. This also the very most polysaccharide sulfatides compounds produced by the several macroalgae, like the brown macroalgae, Sargassum turbinaria. They have some supported polysaccharides such as fucoidan, carginin, and ulfan also reported have activity against several cell lines, including the tumors. In this study, we use 16 specimens of the macroalgae collected from the easterns, especially from the Tua, from the Kupang, also the Maluku. And after we extracted using ethanol for 20 hours and washing 2 times, after that we tried using freeze dry, and we do for the cytotoxic assay using MDA, MB2 31 cells of the breast cancer cells. And after 24 and until 48 hours, the cell of viability was observed. And this is the results from the extractions. We know that Turbinaria ornata from the Tua has the highest yield of the ethanolic extracts, with the most 9% of the grades, total grades biomass. And for the cytotoxic assays, 3 of the specimens or 3 of the species have shown a cytotoxic against MDR, MD31s, the hypnea. This is the doxo, or the positive control of the cancer drugs, doxorubicins, with the concentration 2 microgram per milliliters. And we know hypnea, also Chaetomorphosa and Turbinaria, with the concentration of 1 microgram per milliliters, have the cell viability less than 100%. And this is the profile of the cells counters. And from this data, knows that in 100 microgram per milliliters of the extracts, they have the cell viability percentage of cell viability less than 100%, with the species of Caulerpa, Racemosa, Larynxia intricata, Galaxora, Sargassum, Chaetomorphosa, Classa, Hypnea, and Turbinaria ornata. But this we choose three of the species because this Turbinaria ornata have the high yields, also show the activity. So the 3 of the species is the potential to further investigations. So from the conclusions from this work, we choose 3 of the species, Turbinaria ornata, Hypnea, and Chaetomorphosa, with the IC50 less than 100%, 100 microgram per milliliter. We will investigate for the further investigations, such of the looking for the active compounds, also the detail of the assays. Thank you for your attention.
Okay, thank you very much for all 3 presenters. Now I would like to ask all 3 presenters to put their video on, please? Okay, thank you. Now then, I haven't got any question on the chat room, so I would like to invite our audience to raise their hand if there is something need to be clarified to all three presenters. Is there any? We still have plenty of time. We have 12 minutes to do so.
Can I ask Dr. Sugiono for?
Oh yes, please, Bu Yatri Absari, the speaker would like to ask, and please, if to whom your question will be directed to.
Yeah.
Okay, the time
thank
is
you
yours.
for the opportunity. I'd like to ask to Ibu Tuti, since it's related with my research about Cholera pallidolifera. I see in the Bu Tuti presentation that Cholera pallidolifera has anti-cancer activities, isn't it? Yeah. Then I would like to ask if I missed the slide before, and how to extract the Cholera pallidolifera, and how the anti-cancer activities in Cholera pallidolifera, and from what region did you collect the Cholera pallidolifera? That's all. Thank you for your opinion.
Okay, there are plenty of questions that's been raised. So is there contain anti-cancer, from which region, what the method you use to extract? Okay, Bu Tuti, the time is yours to respond to three Absari question.
Thank you, Pak Sugiono. Thank you, Ibu Yatri. Can you hear our voice?
Yes, I can hear your voice, Bu Tuti.
Yeah, thank you for your questions. Actually, for the cholera, we also have the activity, but in this data, why we choose only turbinaria, Chytomorphosa, and Hypnea. Actually, the cholera in this data, the cholera activity is not significant. It means if the concentration is higher, the cytotoxic is less. So we didn't choose this or a species to further investigations. And we also know that the yield of cholera is not so high in ethanolic extracts. And how we the extracts we tried the first we tried seaweed is washing with the waters, and we re-dried again in the oven. After that we macerate with the ethanolic extracts. We choose 100%. I mean, it's the absolute ethanolic extract. And after that, we tried using the freeze dried to going to the cytotoxic cells, because we removing the water's contents, also the minimize the salt.
Okay, I would like to add Bu Yatri question actually. Do you think that the extraction method you use actually affected the result that you got? You know, but do you think that a different extraction method might have been more beneficial in terms of finding anti-cancer, substance from the sample that you do?
Yeah. Why we choose the ethanolic extracts, because we just looking for the if the seaweed is the potentials, ethanol solvent is more acceptable by the regulations, if we will go into the next application sites like this.
Okay.
Because we know that seaweed actually is not so active for the anti-cancer compared to the other marine biota, but they have if the comparison to the cytotoxicity, it is the more edibles going. So the target is maybe for the functional food or something like this.
Okay, thank you very much, Buyatri and Pututi, for the question and answer. We still have another 7 minutes to do some discussion. Is there any question from the audience? If not, I would like to ask to Pak Nanang actually. You mentioned in your finding that the Cartesian isolate actually showed the lowest lipase activity. Do you think it is due to the percentage of Cartesian within that extract or within the Cartesian isolate, or is there any other mechanism that you can provide to us? Why the inhibition or the represence of the enzyme is such low in comparisons to the other two treatment that you use. And also you saw that the lipase activity was such low. Would you mind, do you have any idea, is it the affected both LDL or HDL and HDL cholesterol, or is this specific for a certain cholesterol? Okay, that's my simple questions for Pak Nanang. You still mute, sir. Would you mind to unmute?
Sorry. Thank you, Mr. Sukiono, Dr. Sukiono. And this the result, Cartesian isolate, but no lowest activity, but lowest IC50.
IC50, okay.
Lowest activity, highest activity. Yeah. And the mechanism of lipase inhibitor lipase activity is specific for total cholesterol and LDL in the prophylactic.
Okay, well, that's very promising then. So we can use that. I think in the ethnobotanical point of view, your gambir is actually frequently used for in Japanese custom for you call it minang, makan sirih, that's use that. I think quite good actually. Thank you, Pak Nanang, for the response. Is there any other questions you might ask from the audience? Feel free to do so. If not, I might use the time to explore Pak Nanang. After completing or after presenting your finding, what's your next experiment that you're going to do, and what sort of result that you are expecting actually?
Okay. This research part of global research in my institution, and in this research I do preclinical study, especially in vitro study. But the next experiment and research I follow to preclinical study in animal study, and the goals of this research of phytopharmaca product for Uncaria grambir to anti-dyslipidemia product.
I think the host, would you mind to mute Bu Ruth Martawini, please? Sorry. Okay. Thank you for the response, Pak Nanang. It's quite promising, actually. So it will be very useful for somebody like me who's got a problem with, hyperlipidemia. So that's good. Okay. Is there any question from the audience? We still have like 2 minutes to proceed in this discussion. Well, it seems that everybody has been satisfied and happy with the presentation and finding being said to us. So I would like to conclude this session by wishing you all three researchers to go further, and good luck with the next stage of research. And I wish you all stay happy and healthy in the future. Okay, thank you very much for this presentation, and I give you a big applause for that. Okay, thank you. And then now I would like to return the sessions to Pak Naro. Pak Naro, still got 1 minute left, but we have succeeded, carrying out this session. So Pak Naro, the time is yours. Thank you very much, and I would like to apologize for any inconvenience it may have caused. Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Pak Naro, the time is yours.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Sugiono, for hosting the sessions. What a very interactive sessions. Bapak Ibu, we'll have a break until 4 p.m. We'll have a break for 10 minutes at your own home. Yeah, you may have a coffee or tea for 10 minutes. Yeah, please be returned to the sessions.
Pak Naro, you may say that the coffee is available in the next room. Please help yourself.
Yeah, Bapak Ibu. Yeah, please enjoy your 10 minutes break. Thank you so much.
Thank you. I'm Pak Naro, thank you very much. I may leave the session. Thank you.
Okay. Thank you.
Hello, good evening, Bapak Ibu. Welcome back to our sessions, parallel session 3 in room 3. And I would like to invite next moderator, but before that, let us see some video from our partners from PT NutriLab, please. Thank you very much, Bapak Ibu. And now let me introduce for the next moderator, Dr. Linda, and let me introduce her background a little bit. So Dr. Linda Sukmarini right now is a researcher in the Research Center for Biotechnology, National Research and Innovation Agency, Republic of Indonesia, BRIN. And she was graduated in biological chemistry, peptide engineering from Berlin International Graduate School of Natural Science and Engineering. And her research is about microbial natural product-based drug discovery. And Dr. Linda Sukmarini, are you there? Are you ready? Okay, so please. I'm sorry. Mute. Okay, so please, Dr. Linda, time is yours.
Okay, thank you for your kind introduction, Dr. Penny. Okay, well, distinguished presenters, guests, ladies and gentlemen, good afternoon. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to welcome you all to this parallel session 3 today. Thank you for stay tuned in this last session. So in this occasion, we would like to have 5 presentations from Ms. Azra Zahra from the Center Biology BRIN. After that, we will have a presenter from Department of Biology, University of Bengkulu, from Rizky Hadi Wibowo. And then we continue to the next presenter from Department of Food Science, University Kebangsaan Malaysia, Hafiz Binti Abdurrahman. And then we also have a presenter from industry, Ms. Fitria Dwi Ayuning Tias from PT Wadya Primamulya, and last but not least, Ms. Rismawati Rashid from University Muslim Indonesia. Okay, before the presentation begins, let me confirm first that all the speakers are already in this room.
Yes, I have.
Okay, okay, then okay, thanks. Okay, I think there's all there. And then I will again highlight the procedure of the session. So we will have 2 parts, presentation session and Q&A session at the end. So each presenter will have 10 minutes pre-recorded video. Hopefully this will be in time frame. And I hope for all the speakers to activate their camera on during the pre-recorded video presentation as well as Q&A session. So questions from all the participants are delivering used by Q&A, and if necessary, also you can raise your hand and give the question directly to the corresponding speaker. But don't forget to mention your institution and to whom you deliver the question. Okay. So without further ado, I would like to welcome first the first presenter, Ms. Zahra Nadira from Center for Biology, BRIN. Are you ready, Ms. Zahra? Okay. Are you there? Okay. Yep.
Assalamualaikum warahmatullahi wabarakatuh. On this ISIBIO 8, I am Azra Zahra Nadira Ikhwani from the National Research and Innovation Agency, BRIN, which formerly I was from the Indonesian Institute of Sciences, LIPI. Representing my colleagues will present our study entitled Investigation of Cellulosic Yeast from Soil and Leaf Litter of Savanna in Kupang, East Nusa Tenggara, Indonesia. Introduction. Lignocellulosic biomass (LCB) on Earth is very abundant, derived from plant biomass, including agricultural waste, energy crops, woody residue, and human waste. Lignocellulose is a component of plant cellulose, which is mainly composed of cellulose, hemicellulose, and lignin, making a complex assembly of polymers. The components of LCB, with their abundant availability, are the main attraction to make them raw materials in various chemical and biotechnology industrial production. One of its uses is as substitute for non-renewable fossil energy fuels, which can be called biofuels or bioenergy, such as producing bioethanol. The hydrolysis step is required for the conversion of biomass into monomeric sugars, for subsequent fermentation into products such as bioethanol. Hydrolysis can be acidic or enzymatic. The enzymatic method has several advantages over the use of acids. The enzymatic hydrolysis of cellulose is catalyzed by a class of enzymes called cellulase. There are three types of cellulase. The first, endoglucanase, which breaks the bonds in amorphous region, and then exoglucanase breaks down bonds in crystalline region, and cellobiose hydrolyze cellobiose to glucose. Endoglucanase activity that distinguishes from exoglucanase has been generally studied on carboxymethyl cellulose or CMC substrate because of its identical in chemical structure to insoluble cellulose. From this, the fermentation for bioethanol production is started. The source of this enzyme can be obtained from microorganisms such as yeast, which are known to have been used by various types to produce alcohol. And in this study, we are interested in isolating and screening potential cellulolytic activity of yeast from savanna ecosystems. Because their geographical features characterized by vegetation growing under dry and hot conditions can become hotspots for the discovery of potential yeast with a high possible ability to degrade plant material. And Indonesia, as one of the world's mega biodiversity, has a variety of ecosystems, including the savanna in the Kupang area, East Nusa Tenggara, which is included in the Wallace area, which is known to have various endemic species of plants and animals. The method used for yeast isolation was leaf litter and soil samples added with physiological saline and twin 80 solution, which was then shaken at 120 rpm. 200 microliter of the suspension was grown on Los Bengal Coral Venicle agar RBCA media. The grown yeast were purified and identified molecularly using 26 srDNA. And then the yeast isolate obtained were screened for their cellulose ability by inoculating the yeast cell into perforated carboxymethyl cellulose CMC agar medium. And then the Congo red dragon was put into the growing colonies. Formation of cliff zone around the colonies indicate hydrolytic activity, and ratio of cliff zone to colonies areas was calculated as cellulolytic index. And last, we selected the isolate, then measured their endoglucanase activity using CMC substrate on the effect of pH, nitrogen sources, and temperature. Measurement will carry out using the reducing sugar method using dinitrosalicylate acid or DNS. The results we obtained were 20 isolates of yeast isolated from leaf litter with a total of 11 and 9 from the soil. 10 isolates belong to the phylum Ascomycota and the other 10 belong to Basidiomycota. The highest cellulolytic index was shown in isolate AK10, Pseudozima aphidis, belonging to the Basidiomycota. The value was 3. That value was not significantly different from AK5 and AK6 Candida tenuis with a value of 2.9. In addition, from the 20 isolates obtained, the most identified species were Rhodosporidium paludigenum, AK1, AK2, and AK9. To test the activity of endoglucanase on the effect of media conditions from pH, nitrogen source, and temperature, we continued on the isolates of Candida tenuis AK5 and AK6, and for the comparison was Rhodosporidium paludigenum AK1. The effect of temperature showed that the highest endoglucanase activity was Candida tenuis AK5 with a value of 8.7 unit per milli at an incubation temperature of 35 degrees Celsius. This value was not significantly different from the AK6 isolates of candidate tenuis, and the effect on other temperature tested was also not significantly different on average in this species. The effect of nitrogen sources showed that the highest endoglucanase activity was Candida tenuis IK5 in yeast extract, with a value 8.6 unit per milli. Between the two nitrogen and AK6 in peptone. Both in the Candida tenuis isolate, the value was not significantly different. Meanwhile, the isolate Rhodosporobium paludisgenum AK1, the highest value only shows a value of 6.2 in yeast extract, similar with AK5 in yeast extract. Generally, the enzymatic activity using organic nitrogen sources from yeast extract and peptone were higher than the enzymatic activity using inorganic. Besides temperature and nitrogen sources, pH also plays an important role in the enzyme characteristic. The present study showed endoglucanase activity of isolates Candida tenuis AK5 and 6 reached the peak of the activity at the pH 7, 8.7 and 8.9 unit per milli, while the activity of Rhodosporidium paludisgenum AK1 showed optimum activity at pH 6 with a value 6.2. Endoglucanase activity on the effect of pH of the two isolates of Candida tenuis showed a higher value than Rhodosporidium paludisgenum. And for the conclusion, a total of 20 cellulosic yeasts were isolated from leaf litter and soil of savanna in East Nusa Tenggara, Indonesia. And each of the 10 isolates will belong to Ascomycetes and Basidiomycetes yeast. The isolate AK5 and AK6 showed promising cellulosic yeast, both were identified as Candida tenuis. The highest cellulosic activity of isolate AK5 and AK6 was observed at 8.9 unit per milliliter and 8.7 unit per milliliter. Okay, with optimum temperature 35 degrees Celsius, using yeast extract and peptone as nitrogen sources respectively, and pH 7. The study reported the potential of yeast isolated from Indonesia, especially from leaf litter and soil of savanna. However, the utilization of those isolates for bioprocess, especially on their application in processing agricultural waste for hydrolytic enzyme production, needs further investigation. And thank you for the opportunity and attention. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you for your nice talk, Ms. Zahra, for your talk about cellulosic yeast. So now we are moving to the second speaker, Mr. Rizky Hadibowo. Are you ready, Mr. Rizky? Okay.
Yes, I'm ready.
Please, yeah. Okay.
Assalamualaikum warahmatullahi wabarakatuh, and good afternoon to all. Good afternoon to the moderator, to the committee, participant, and also the other presenters here. Thank you so much for the opportunity that has given to us to share about our informations about our research title, Antibacterial Activity and Phytochemical Analysis of Ethyl Acetate of Micrania micrantha Kunz leaves from Bajang Lebong District, Bengkulu Province. Me as the main author, Rizky Hadibowo, and also our team member from Department of Biology, Faculty of Mathematics and Natural Sciences, University of Bengkulu. So as our introductions, we know that the plant of the Micrania micrantha leaves is one of the most invasive plants in the world. They're very easy to grow in various conditions and considered as a weed to farmer. And also in Baraman's village, Rojang Lebong District, Bengkulu Province, this plant is very easy to grow. So from the information that these plants consist of very high phytochemical compounds, for example, like the flavonoid, alkaloid, tannins, steroids, saponins, and also terpenoid, which is that They consist or they can be suggest as the antibacterial to inhibit some of the human pathogenic bacteria, for example, like the Staphylococcus aureus, Bacillus subtilis, Escherichia coli, and also Pseudomonas aeruginosa. As our experimental methods, so we do several steps. First is lip samplings and symplesia preparations, and then lips extractions, and then phytochemical analysis, and then preparations of the extract concentrations, determinations of the minimum inhibitory concentrations, and also the antibacterial activity assay of Micania micrantha lips extract. So as the result and discussion from this table, we can see that the phytochemical analysis of the Micania micrantha lips extract, the tannin and phenol are positive consist in the extract of the lip based on the ethyl acetate extract. But the other one, for example, like the flavonoid, alkaloid, steroid, and then saponin, terpenoid, and quinone are negative result. So this is the figure of the phytochemical analysis based on the ethyl acetate extract of Micania. So after that, we try to determine the minimum inhibitory concentration, or the MIC, from the extract of the Micania micrantha lips. So we start from the lowest concentrations of 10% concentrations until 100% concentrations. And then we will test them to the human pathogenic bacteria that we use, which is the Bacillus subtilis, Ecology, Pseudomonas aeruginosa, and also the Staphylococcus aureus. So from this table, we can see that the minimum inhibitory concentrations of the lip extract of Micania are very diverse or very different based on the pathogenic that we use. So the minimum inhibitory concentration is about 20% until 40%. After we find the minimum inhibitory for each of the leaves, and then after that we can see here that we want to see about the antibacterial activity of the Micania leaves based on the minimum inhibitory concentration. So we use several another concentrations, and then we test again to our pathogenic bacteria, to Bacillus subtilis, Escherichia coli, Pseudomonas aeruginosa, and also Staphylococcus aureus. So from this table, we can see that the best concentrations produced by the lip extract are very diverse. So it depends to the bacteria that we use. So for the Bacillus subtilis, the best concentrations, so the largest diameter of inhibition is about 45 concentrations, and then for the E. coli in 35 concentrations, and then for Pseudomonas aeruginosa in 35 concentrations, while the Staphylococcus aureus in 55 concentrations. And then the category for the inhibition zones is about moderate, and then several of them are strong. So from the discussion, we can see that the presence of the inhibition zone from the ethyl acetate extract of Micania micrantha leaves is also influenced by the content of its phytochemical compounds, for example, like the tannin and also phenol. So we know that the tannin and also phenol is an organic compound that have the activity of the antibacterial. So the conclusion is the result of the antibacterial activity of the ethyl acetate extract of Micania micrantha leaves from Baromanis Phyllis Rejang Lebong Regency were able to inhibit pathogenic bacteria, for example, like Bacillus subtilis, E. coli, Pseudomonas, and also Staphylococcus aureus. And then based on this study, the Micania micrantha leaves extract may be can be the alternative in the medical world as the treatment for the infections caused by human pathogenic bacteria. And then for the further studies, gas chromatography mass spectroscopies or GCMS analysis can be carried out to determine the main compound from the Micania micrantha leaves, which truly goes as the antibacterial. And furthermore, it's important to know the side effect of Micania micrantha leaves extract in people. So I think that's all the information about our research, and thank you so much for the attention. We bring back to the moderator.
Okay, thank you, Mr. Rizky Hadibowo. Okay, next we will have Miss Hafiz Binti Abdurrahman from University Kebangsaan Malaysia. Are you there, Miss Abdurrahman? Okay, now enjoy the recorded video first. Please, the committee. Okay.
Good day, everyone. I am Hafiz Abdurrahman from Department of Food Sciences, University Kebangsaan, Malaysia. First of all, I would like to thank the organizer for giving me the opportunity to share my latest findings on metabolite profiling and bioactivities of Malaysian black turmeric extract using NMR-based metabolomics approach. So why NMR? It is because NMR is a powerful tool that can be used to elucidate structural information and also isolate the metabolites within a complex system. Both NMR and also multivariate data analysis have been used widely for metabolite profiling and for the determination of differences between sample as well as characterization on the composition of various types of plants and also biofluids. So in this study, the variability in the chemical composition and also discrimination among the different extract of black turmeric was elucidated using NMR-based metabolomics. Apart from that, we also determined the correlation between metabolites and biological activities. So curcuma cassia, as we can see on the left side here, is known as black turmeric or kunyit hitam in Malaysia. It is a medicinal plant that is native to tropical South Asia. So the rhizomes of the plants have a bluish purplish ring on its rhizome, and it also have a purplish color on the leaves itself. The plant is also have been used traditionally to treat asthma, bronchitis, cold, fever, wounds, and gout. In addition, this plant also have been reported to possess antibacterial, antimicrobial, antifungal, anti-inflammatory, anti-cancer, and also antioxidant effects. So chemical compounds that have been reported before previously includes tannins, flavonoids, alkaloids, and also phenols. So plant metabolites are made up from both polar and also non-polar compounds. In order to develop the plant herb or plant-based nutraceutical products, it is very crucial that we extract the compound efficiently. So how to two factors that is important is drying techniques or processing techniques of the herbs, and also the selection of the solvent. So this is because it will affect the amount and also the type of extracted compounds from the plant matrix, which will reflect in the bioactivities of the plant extract. So we have two objectives in this study. First one is to determine the effect of drying techniques, which we use two different drying techniques, freeze drying and oven drying, and extraction solvents on the phenolic content, antioxidant, and anti-hyperglycemic activities. The second objective is to determine the correlation between the biological activities of curcuma cassia extract and the metabolites using NMR-based metabolomic approach. First, we started our study with drying the sample. Rhizome and leaves were subjected to different drying technique, which is freeze drying and oven dry. And then the dried sample will grind into powdered form, and we extract them using different ethanol to water ratio. Here we have 0-100, 50-50, 80-20, and 100-0. So 0-100 means water extract, 100 zero means that is ethanolic extract. And then we remove the solvent using rotary evaporator at 40 degrees Celsius. We measure the TPC, the antioxidant test by DPPH and FRAP, the anti-hyperglycemic test using alpha-glucosidase inhibitory activity, and then we measure the metabolites using proton and 1D proton NMR and also 2D JRES NMR. We also measure the correlation using SIMCA software for multivariate data analysis. So here we can see that freeze dry is better than oven dry in terms of drying, and in terms of solvent, 100% ethanolic extract is better than the other solvent. As we can see, the total phenolic content is highest in the freeze-dried rhizome extract of 100% ethanolic extract compared to the others. So the same trend can be seen here in the FRAP assay, whereas the rhizome freeze-dried ethanolic 100% ethanolic extract showed highest FRAP activity. So for the DPPH, also we can see that rhizome freeze-dried extract showed lowest IC50 value, both 100% and 80%. In terms of value, even though 80% is lower in IC50 value, but it is not statistically different. In terms of alpha-glucosidase inhibitory activity, also the same trend, whereas the rhizome freeze-dried extract showed lower IC50 value, both 80 and 100% showed low IC50 value, even though the 100% ethanolic extract showed lower IC50 compared to 80, but they are not statistically different. They are the same. So here we have on the left side is the full spectra of the different freeze-dried curcuma cassia rhizome extract. So we just proceed with the rhizome extract of the freeze-dried because it is the most active. So we can see that they are different, especially in the aromatic region. Here is 5.5 to 9.0 ppm. And 80% and also 100% here showed a lot of metabolites in the aromatic region, whereby the phenolic compounds are. It also in PCA score plot, it also show a clear separation between extract without any significant outlier. So we can see here, this is 100% and 80% is here, 50% and water extract is here. So 100% is clearly separated from the others. So these are all the 27 metabolites that we identified in all the 4 different extracts. So in terms of the next thing is we analyze the PLS by plot to identify what is the compound that is responsible or what is the compound contributing to the bioactivities measured previously. So the 100% extract here, the yellow rectangle showed the yellow triangle dots on the upper right quadrant, which is very close to the red star here. Okay, the red star represent the bioactivities measure, so it is very close to each other. It suggests strong correlation between them. So for the water extract and 50% extract were located in the lower left quadrant of the plot, which were far from the bioactivity, which indicate a very low correlation within the reaction variables. So the green dots here, the small green dot here represent the NMR signal and the metabolites. So majority of the metabolites you can see are located near to the 80 to the 100 and also to the 80% extract. So it showed that this 100% and 80% solvent is the best solvent to extract more compounds compared to the water and also 50%, whereby only small amount of metabolites are extracted and detected by NMR. And also there are 2 compounds that are responsible for the bioactivities and that are high, very high in the 100% extract, which is compound number 4. This is the name of the compound is quite long, and the compound number 9 here, which is the cordion. So these are two very significant compound that is responsible for the high antioxidant and anti-hyperglycemic activities in 100% ethanolic extract. So for conclusion, based on the result, it was concluded that 100% freeze-dried rhizome extract showed higher TPC and FRAP activity, whereas for DPPH and also for alpha-glucosidase inhibitory activity, both 100% and 80% showed lowest IC50 value. So in terms of overall, I would say 100% is the best solvent, and freeze dry is the best drying methods. So two metabolites that were identified is higher in the 100% extract is number 4 and number 9. So and this study have to provide some guidance on the development of some nutraceutical or functional products from Curcuma cassia extract. With that, thank you for listening and have a great day.
Okay, very nice work from Ms. Hafiza. Okay, now we are moving on next presenter, Ms. Fitria Dwi Ayuning Tias from PT Wadya Primamulya. Time is yours.
Hello, everyone. Let me introduce myself. My name is Fitria Dwi Ayuning Tias. I represented PT Wadya Primamulya Jakarta and also Research Center of Nanoscience and Nanotechnology, ITB Bandung. And today I will talk about bioimaging application in stem cell behavior and mechanobiology. So as an introduction, as we know that stem cell research and stem cell engineering research has rapidly increased in recent years and very popular because of its potential as a source for regenerative therapy. As we know that there are 3 types of stem cells we can obtain from the human body. The first is embryonic stem cell from the embryonic stage. And the second is adult stem cell from the tissue, the adult tissue and the adult cells. For example, mesenchymal stem cells with its multi-potential. It's very popular for regenerative therapy now. And it can, because it can easily isolate it from the adult tissue such as bone marrow and then adipose tissue and also the umbilical cord. And the third one is induced pluripotent stem cell, the one that reprogram the adult cell, any kind of adult cells, for example, skin or hepatocyte or any adult cell, and reprogram to become as potential as the embryonic stem cells. And the stem cell isolated from the human body, although that's adult stem cell or iPS or embryonic stem cell with its potential to become to differentiate into specific cells, then cultivate and been through an expansion process and then differentiate into specific differentiated cells, and then after that were used for the regenerative therapy. And the differentiation process is very important process. And in the recent years, researcher has been developing the way to differentiate the stem cell to become the specific cell lineage. And there's a lot of ways, such as adding the soluble factors, growth factors, or even changing the biophysical and microenvironmental cues to help the cells decide its fate. And in our group research, we modify the microenvironmental cues for the cells to regulate the stem cell fate. And the cells will sense the modification of the substrate and then can transfer the signal into the intracellular part during mechanotransduction phenomena. And after that, the cell can change its cellular function and also help it to decide which way or which direction they want to go to differentiate. So we call it the cell-fate decision. And it's mainly because of these two phenomena, the cell-cell interaction and cell-substrate interaction, that help cells to sense what happened in the microenvironment. And this is cellular property which play a role in mechanotransduction, such as extracellular matrix, integrin, focal adhesion protein, chromatin, cation junction, and F-actin, the cytoskeleton, and also nuclear ligand. These all cellular properties, the changing in the cellular properties and morphology occur during mechanotransduction, and this is what we always observe, and qualitatively or quantitatively. One techniques in our group we usually use to gain more understanding about the mechanotransduction phenomena in stem cell engineering is bioimaging. Bioimaging is a non-invasive process of visualizing biological activity in a specific period. It's often used to gain information on 3D structure of the specimen from the outside without physical interference. Bioimaging also includes the methods visualizing biological material that has been fixed for observation. And in our research group, we usually use bioimaging to understand cell morphology, structure, 2D or 3D, protein localization, morphogenesis, spatial heterogeneity, dynamic cell interaction, and cell apoptosis. And this is the method. We observe the cellular properties, both using live cell imaging or fixed cell imaging. For live cell imaging, this is the example. We usually use a time-lapse observation and cell tracking. And for fixed cell imaging, we usually use the immunofluorescence staining. So this is the example result. And the live cell imaging usually used its function is to understand the cell behavior and the alteration of function, while fixed cell imaging usually to understand the cell structure, the protein localization, something like that. And this is two examples. So I would like to show several results our group have. This is a time-lapse of observation. So from this method, we know that time-lapse of observation could be used to understand cell behavior as a response to stimulation. Like in this result, we changed the substrate. We have three different substrate, and then we cultivate the mesenchymal stem cell above it, and we found that the cells respond differently for each substrate. So by time-lapse observation, we could understand the changing of cell behavior in response to a stimulation. And then this is a fixed cell imaging using immunofluorescence staining and observed by confocal laser scanning microscope. And this kind of results let us know that immunofluorescence staining also could be used to understand the cellular protein localization, like in this case, the localization of vinculin towards F-actin. On the other hand, we also can visualize the 3D structure of the cells using a confocal laser scanning microscope and immunofluorescence staining as a techniques in bioimaging. And for example, in this result, we construct the 3D structure of stem cell, human mesenchymal stem cell aggregate through observation using confocal laser scanning microscope. And then, next. The other things is the optical sectioning in confocal laser scanning microscope observation result could provide a comprehensive data and deep understanding of cellular structure and mechanotransduction phenomena like this within cells in response to stimuli. We can see the apical, basal, and exit section, XY section through this bioimaging technique. And it gives a more understanding and comprehensive data to understand the phenomenon. So as a conclusion, the bioimaging technique is very important to use and to elucidate microbiology phenomena in stem cell engineering. And this technique also needed to gain understanding in cell research, especially to observe these several parameters. And also it required to provide more reliable and proven data in life science research. So that's all my presentation. Thank you very much. Thank you for your attention. See you.
Okay, well, very nice presentation from Ms. Fitria. Now, for the last presenter, we will have Ms. Rismawati Rashid from Universitas Muslim Indonesia. Okay, she will talk about the effect of hydrochloride nickel. Okay, now ready.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity given to us. I would like to explain about the research that we have carried out with the title Effect of Hydrogen Chloride Nickel Gamma Alumina Catalyst on Biofluid in Palm Oil Catalytic Cracking. The authors are Rismawati Rashid, Muhammad Panji, Eko Rusdi, Rahmania Malik, and Darnesi, the all authors from Faculty of Industrial Technology, Universitas Muslim Indonesia. Introduction. Fossil fuels are non-renewable raw materials, so their supply is reduced due to high use compared to fuel supplies. The second, alternative fuels are needed to substitute oil fuels such as biofuels. Biofuel production can minimize the use of fossil fuels, then their use is more environmentally friendly. Biofuel, the raw materials of vegetable oils, as a source for biofuel include palm oil and others. One of biofuel reaction process is catalytic cracking. Catalytic cracking is a reaction that cuts long hydrocarbon chains into shorter hydrocarbon chains. The uniqueness of this reaction is that the hydrocarbon molecules are crushed randomly to produce a mixture of smaller hydrocarbons. Biofuel consists of several types of fuels such as gasoline, kerosene, and biodiesel. There are three steps in research methods. The first, catalyst preparation. Impregnation process in preparation. Nickel nitrate into the gamma alumina catalyst support was performed by adding hydrogen chloride 0.4 normal about 10 milli into the nickel gamma alumina catalyst. Then the mixture was dried for 8 hours at temperature of 110 degrees Celsius. Afterwards, the mixture was calcined 650 degrees Celsius for 3 hours. The second step is catalytic cracking reaction. Palm oil with a volume 50 milli was input into a bed catalytic cracking reactor. The catalyst used was 5% of the volume of the material. The reaction temperature were 330 until 380 degrees Celsius, respectively, with a reaction time about 2 hours. And the last step in analysis catalyst is XRD, X-ray diffraction, and analysis product is GCMS or gas chromatography, mass spectrometry. Result and discussion. XRD analysis of catalysts have diffraction peaks 2 theta at 37 derajat, 39, 45, 66, and 67 derajat. Based on International Center for Depression data, gamma alumina has a peak at 2 theta 45 and 67 derajat. The nickel oxide perform is at the peak position 2 theta is 37 derajat, is based on ICCD. The position of hydrogen chloride is estimated to be at 2 theta is 39 derajat with the resulting intensity of 10.557%. The position of hydrogen chloride and nickel on the catalyst support result in an intensity that almost matches the preparation process. Yield biofuel. According to the graphic, palm oil has not been converted into biofuel at reaction temperature below 350 derajat Celsius, indicating that the activation energy is quite high. It can be concluded that the temperature has a significant effect on the conversion of the reaction. This is relevant to the research conducted by ASIS et al. 2019. The study resulted in the highest biofuel yield on 64% at temperature 380 derajat Celsius. Previous research conducted by Budianto et al. 2019 result in a yield of 26.40% weight in catalytic cracking RBD stearine with zinc 8 seed SM5 gamma alumina catalyst at temperature 450 derajat Celsius. Biogasoline obtained from the catalytic cracking of palm oil consists of C5 until C11. The percentage of gasoline 11.79% as presented at table. The biogasoline compounds include cyclopentane, benzene, cyclohexane, octane, tetradecan, pentadecan, and others. And the second product, biokerosene. The catalytic cracking of palm oil produces biokerosene C12 and C15, which consists of 4 types of biokerosene as presentable. The percentage of biokerosene in the biofuel products from this research is 27.06%. And biodiesel product, the catalytic cracking of palm oil using hydrogen chloride nickel gamma alumina catalyst produce biodiesel C16 until C20. The types of chemical compounds of biodiesel produced from the reaction are 6 types as present at table. The percentage of biodiesel in the biofuel from the reaction is 44.74%. Compare the performance of catalysts. There are 2 catalysts, hydrogen chloride nickel gamma alumina and hydrogen chloride without nickel and support gamma alumina. The all catalysts produce 3 types fuels like gasoline, kerosene, and diesel. And based on the graphic, it can be concluded that the catalysts that use nickel or without nickel produce a more dominant product to biodiesel. Conclusion, the catalytic cracking of palm oil indicates that catalyst and reaction temperature can affect the yield biofuels. The reaction temperature in the range 350 until 360 degrees Celsius, obtain biofuel yield on 12.8 until 39%, and at temperature 380 degrees Celsius, the biofuel yield increased to 64%. The obtained biofuel contains gasoline 12.79%, kerosene 27.06%, and biodiesel 44.74%. The last has hydrogen chloride and nickel gamma alumina catalyst, more selective towards biodiesel. I close this presentation with a thank you. Hope it is useful. Thank you very much.
Okay, thank you, Ibu Rismawati Rashid. Okay, now, all right, now we come to the question and answer session. We still have like 8 minutes around, and then I would like to invite all the audience to raise their hand and deliver the question for one of the speakers. Yes, please. If you have. Okay. Okay, Mbak Penny.
Thank you very much, Mbak Linda. Actually, I have a questions for Dr. Fitria. I saw your results. It's about G1, G2, and G3, right? But I'm not really clear what is the difference between G1 and G2 and G3, actually. And did you do fluorescence imaging live in a live cells, or in you preserve it first and fix it first, or something like that? And what is the difference between G1, G2, and G3, and what did you do with them? Because actually I'm not really sure I'm not really know about the stem cell, but I'm really interested in stem cell. Thank you very much.
Thank you very much for your question. So I would like to answer about your question. So in my research about using G1, G3, and G5, there's a modification using nanotopography substrate. So I call it G1, G3, and G5. It's because different generation. So I'm using, I call it dendrimer surface. That's a nanotopography modification substrate. So if G1 is only one generation, and then G3 is like 3 generations, so 3 steps of nanotopography, 3 layers, such as like that. So it's higher substrate and then higher topography, and G5 is more higher because it's 5 layers of nanotopography substrate. And the stem cell I culture in the different types of nanotopography substrate, G1, G3, G5, and then the stem cell responds differently. So the behavior of stem cell changing as a response to the nanotopography modification. So that's I can explain like that about the G1, G3, G5. I hope it's clear. And about the staining technique. So for the time-lapse observation, I didn't do any staining, so it's a live cell. I observe using the live imaging instrument for 10 days without any staining. And then for the confocal laser scanning microscope observation, we fix the cells. So we fix using immunofluorescence staining, usually using the F-actin antibody, and then lamine or DAPI, and then we observe using confocal laser scanning microscope.
Okay,
so,
great. But one more question, but which one is better between G1, G3, or G5? It's different layer, right?
Yes,
yeah.
yes.
Which one is better depend on according to your purpose?
So in my purpose, I would like to differentiate the human mesenchymal stem cell into cardiomyocyte, cardiac muscle cell, and it goes with the G5 substrate. But G1 and G3, it goes to different lineage of differentiation. So G1 goes to smooth muscle cells differentiation, and G3 goes to muscle cells differentiation. So it different types of muscle cells. But as my purpose is to find cardiomyocyte, cardiac muscle cells. So G5 is the better one.
Oh, I see. So it's dependent purpose that you want, so you can adjust the number of the layer, right?
Yes,
okay.
Yes,
Thank you very much for your explanations.
Thank you very much for the question.
Okay, thank you, Mbak Penny and Mbak Fitri for nice discussion. And then maybe we have one more question, please. Okay. Yeah, Mas ISIBIO, your time, please.
Thank you, Mbak Linda. Maybe I have question for Ms. Rismawati. Sorry, I missed your presentation, but I just read your abstract. In your study, you want to know the catalyst performance in the biofuel production from palm oil. So in this case, because you did impregnation method for preparing the catalyst, did you do the how to say, like the metal content that leach in your product? That one, the first question. And the second one, because this is the bifunctional catalyst, it has Lewis acid and also Bronsted acid. Which one is responsible for your reaction? Maybe that's the question.
Thank you for the question. I would like to answer this question. Nickel is one of metal catalyst until calcination process is changed to nickel oxide, and nickel oxide is has to cut long carbon hydrogen to produce to produce small hydrocarbon, and then this is three types product. If small hydrocarbon chains which produce gasoline, this is one of functional logam in the reaction. I think it is enough.
Okay, Mas Bayu, do you have any question more? Is it enough for you?
Yeah, I think it's enough, Linda. Thank you very much.
Melinda, I'm sorry. Please turn on your mic, please.
Okay, sorry. Okay, I would like to add more question to Ibu Risma. Ibu Risma, I wonder. I curious how far your research is close to the industry now. For the application. I think it's very promising.
The application this reaction is more because one of industrial like Pertamina to produce biofuel, and now from palm oil with hydrocracking reaction, because hydrocracking reaction is high temperature and pressure, and now maybe the future the reaction is potential to relation with oil with palm oil, because this is effective and efficient reaction, because reaction in low temperature and low pressure. Yes, I think it is enough.
Okay, thank you for your explanation. Okay, unfortunately the time is already up, and then finally we come to the end of part of our presentation. Once again, we would like to say thank you to all presenters for the informative and very interesting talk, and to the audience, and hopefully this presentation will be beneficial for everybody. And I will give the session to Mbak Penny again. Okay, thank you for attention.
Thank you very much. Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mbak Linda and Bapak Ibu. Thank you very much for staying with us until the end of our sessions. And I would like to say for everyone that let's go to the main room, maybe to hear for tomorrow's informations. And let's go there and see you tomorrow. Please go to the main room. Thank you.
Recording in progress. Ladies and gentlemen, welcome back to the main room of ISIBIO 2021. We would like to thank all the speakers and moderator for the insightful session. Now we have come to the end of the first day of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, ISIBIO 2021. We would like to remind you for the second day of ISIBIO tomorrow, we will open the registration at 7:30 and start at 8 a.m. Jakarta time. And for tomorrow, we will have keynote speaker, Professor Duk Jo from Asian Federation on Biotechnology. And plenary session, we have speakers, Professor Zihua Jiang from Department of Animal Sciences, Washington State of University, USA, and Mr. Zahir Ahli, PhD, from Center for Desert Agriculture, King Abdullah University of Science and Technology, Saudi Arabia. And at the end of the symposium tomorrow, there will be announcement of 6 best presenter in ISIBIO 2021. So stay tuned, and we hope to see you too tomorrow. We would like to thank all of you for your participation. And on behalf of the official committee, we would like to apologize for any inconvenient matters during this today event. So see you tomorrow and goodbye.
Thank you, Mbak Wiwit.